Dear colleagues, this is 2:30, and we should start our event, the 13th Central European Symposium. My name is Małgorzata Sznitowska. I'm a head of the Department of Pharmaceutical Technology at the Medical University of Gdańsk. On behalf of the organizing committee and representing the pharmaceutical faculty of the Medical University in Gdańsk, I am pleased to welcome all participants of the 13th Central European Symposium on Pharmaceutical Technology. The series of these events was initiated in 1995 by a group of devoted scientists from Slovenia. We owed the idea of symposium to the enthusiasm of Professor Aleš Mrar, Franz Kociek, Jelka Schmidt-Korbar from University of Ljubljana, as well as to Professor Flavio Rubessa from Italy. The idea was fully supported by their friends, reputed professors, Douwe Breimer from the Netherlands, Dominique Duchan from France, Attila Hincal from Turkey, Alexander Florence from United Kingdom, Hans Junginger from Germany, and Professor Clara Pintiehody from Hungary. I would like to warmly greet all of these persons, thanks to whom we have the platform where academia meets industry, the east of Europe meets west, and south meets north. And now we are really in the center of Europe. More even than during previous 12 editions of the symposium. When 3 years ago I was proposed by Professor Mrar in Szeged, that was a kind proposal to organize next CESPT meeting in Gdańsk. I presented this 2 slides showing that in Poland there is really a center of Europe. After 26 years since the CESPT meetings had started, the symposium is organized in Gdańsk in a new hybrid form. This partly online event was not actually our choice, but is a result of the global situation. The pandemic did not allow us to meet last year to keep the tradition of the 2 years gap between the meetings. However, we are happy that in September 2021, we are able to invite more than 1/3 of the participants to attend the symposium in person, gathering in a traditional way in a main lecture hall of our university. Hopefully, this hybrid CESPT is a sign of recovering from the hard time of pandemic. I would like to welcome warmly all participants present in the lecture hall and the large group of you in front in the computers, being at your laboratories, or some working at homes. I have an honor to welcome Professor Michał Markuszewski, Vice Rector for Science of our university, and Professor Agnieszka Zimmerman, Vice President of the Polish Pharmaceutical Society. With us online is the new President of the Polish Pharmaceutical Society, Professor Bożena Karolewicz. I warmly welcome co-president of the symposium, Professor Rok Drew from Ljubljana, and online is the godfather of the symposium, Professor Aleš Mrar. The honorary committee is also represented by Professor Douwe Breimer, Professor Dominique Duchan, Professor Attila Hincal, and Professor Flavio Rubessa. The sequence of CESP meetings built certainly a background to another initiative of scientific and teaching collaboration in the Central Europe. This is realized for the last years within the technological network called SIPUS-SECA. The leader of this activity is Professor Jelena Parojcic from Belgrade, being present with us online. But also some other coordinators are joining us online, while in Gdańsk are present in person Professor Stane Sercic from Slovenia, Professor Geza Regdon from Szeged, and Professor Andreas Zimmer from Graz. I welcome also all members of the scientific committee, including those who are here in Gdańsk, Professor Beatrice Perisutti and Professor Albin Crystal. Thank you very much to everybody who has accepted our invitation to participate in person or online in our symposium. We are gathering together young and more experienced scientists, all devoted to make research for better medicines. I would like to kindly welcome our eminent lecturers. Let me indicate those who give lectures today. I welcome Professor Breimer, Professor Catherine Tuleu, Professor Maria Vertzoni, and Professor Sebastian Polak. With a great pleasure, I would like to welcome Professor Jörg Breitkreuz from Düsseldorf, who is a president of the APV, the International Association for Pharmaceutical Technology, well known for us because of many initiatives, including organization of the most important pharmaceutical congress in Europe. I believe that besides APV congresses, also CESP has been for years one of the Europe's finest pharmaceutical events. Working on the program of the 13th CESP, we have followed the intention of the previous editions to give pharmaceutical sciences in Central Europe region a platform to bring together colleagues working in the field of pharmaceutical technology, both in industry and academia. The conference provides the possibility for the participants to present their results, discuss new developments, and the future directions of the pharmaceutical technology, manufacturing, and related analysis. The focus of the meeting is on the topics which are already altering the face of pharmaceutical science and industry. These are formulation concepts and delivery strategies for improving patients' compliance, continuous and additive manufacturing, stability and bioavailability of biopharmaceuticals, and biorelevant dissolution testing. The challenges generated by these concepts and new achievements in pharmaceutical technology are addressed by the eminent researchers who are attending the symposium. Thus, the latest research findings will be presented in the form of plenary and key lectures and short talks. Furthermore, a poster session will give the opportunity to exchange technological ideas. Looking forward to have a fruitful conference, we also would like you who are in person to enjoy beautiful city Gdańsk. In Gdańsk, we have three main universities. This is Technical University, University of Gdańsk, and we represent Medical University of Gdańsk. The honorary patron of our symposium is rector of the Medical University of Gdańsk, Professor Marcin Gruchała, who is at the moment at another important meeting. I would like to ask for a short speech our Vice Rector for Science, Professor Michał Markuszewski, Professor is one of our colleagues from the pharmaceutical faculty, head of the Department of Biopharmacy and Pharmacodynamics. On this opportunity, I'll also welcome other professors from our faculty with our deans ahead. So please, Professor Markuszewski.
Mrs. Chair Lady, ladies and gentlemen, on behalf of Honorary Patron of the Symposium, Rector of the Medical University of Gdańsk, Professor Marcin Gruchała, who unfortunately was unable to join us today, it's my great pleasure to welcome you here at the Medical University during the 13th Central European Symposium on Pharmaceutical Technology. Due to the hybrid character of the event, especially welcome all the participants who are present here in person and also all those who are present in the virtual space, which for practically the last 16 months became the only platform for real-time participation in the scientific symposia. It's not a truism to state that the challenges of innovative and generic medicinal products are of utmost importance nowadays. In end of November 2020, European Commission published Pharmaceutical Strategy for Europe, which is a kind of response to the new challenges and situations created after the breakup of the COVID-19 pandemic on health care system in the frames of pharmacy. It's a patient-centered strategy that aims to ensure the quality and safety of medicines while boosting the sector's global competitiveness. The strategy aims to create a regulatory framework that will not easily become obsolete and to help the industry promote research and technology that reaches patients. This is to meet the therapeutic needs while addressing market failures. On the top of priorities listed in the document is high quality, safe, and environmentally sustainable medicines. To achieve this goal, an innovative pharmaceutical technology is a crucial aspect of the drug development. I am sure that this symposium devoted to the challenges of contemporary pharmaceutical technology will be not only a great place to share the results of scientific research, but also a place to exchange ideas of joint cooperation and innovative initiatives. On this occasion, I would like to thank all the sponsors and organizers of this meeting, with particular emphasis on the President of the Scientific Committee, Professor Małgorzata Sznitowska, for their engagement in the organization of the symposium. I am sure that all your efforts put in the organization of the event will result in success, both scientifically and socially, of the conference. I wish all of you enjoy and fertilizing scientific presentations and discussions. Thank you very much.
Thank you, Professor Markuszewski. The second honorary patron and also a sponsor of the symposium is the Polish Pharmaceutical Society. The president, Bożena Karolewicz, head of the Department of Pharmaceutical Technology of the Medical University in Wrocław, is with us online, and in person, I welcome Professor Agnieszka Zimmerman, Vice President of the Society and a pharmacist and a lawyer, head of the Department of Medical Law in our university.
Dear colleagues, please let me read the special letter from Professor Bożena Karolewicz, the President of the Polish Pharmaceutical Society. The Central European Symposium on Pharmaceutical Technology is one of the best symposia on drug delivery systems in Europe, an important event supporting intellectual exchange among members of the international pharmaceutical community. On behalf of the Polish Pharmaceutical Society Executive Board, I would like to address special thanks to the local organizer from the Medical University of Gdańsk Faculty of Pharmacy, President of the Symposium, Professor Małgorzata Sznitowska. Thank you. Co-President of the Symposium, Professor Douwe D from the University of Ljubljana, and members of the honorary and scientific committees. Thank you for coordinating the activities of this important initiative, which is a platform for strengthening cooperation and the valuable exchange and experiences among representatives of the world of science and the pharmaceutical industry. Let the hybrid event form be an occasion for network and brainstorm on experiences in innovation in pharmacy research and formulation concepts. With best regards, Bożena Karolewicz. And from myself, very fruitful meeting. Thank you.
I would like to ask Professor Douwe for a short speech. Professor Douwe from the Department of Pharmaceutical Technology in Ljubljana continues the Professor Mrar's work, being responsible for the series of CESP Symposium and being also the co-president of the actual event.
Thank you for the kind introduction. So, ladies and gentlemen, dear participants of the 13th Central European Symposium on Pharmaceutical Technology, warm greetings also from my side. First of all, I would like to thank the organizers of the symposium, Medical University of Gdańsk, Faculty of Pharmacy, and Polish Pharmaceutical Society for their efforts and perseverance to offer the symposium in beautiful Gdańsk, and for the first time in this CESP conference format also in Poland. As said, the conference has been envisaged to take place in 2020 and had to be postponed to this year due to the troubled times of pandemic. Fortunately, efforts of scientists and mass availability of SARS-CoV-2 vaccines just in time enabled organizers to offer the symposium in a hybrid format and sent an important sign of recovery. As a result, we have roughly 40% of the symposium participants here in Gdańsk, and the rest will participate the conference online. Organizers could have easily followed the more common online format as a current norm, but I strongly believe that online format, although having its advantages like lean execution while still having access to the latest advancements in specific scientific areas, it however lacks an important social component of a proper scientific conference. Conferences are not only about talks, about poster presentations, but also about interactions of people with common scientific interests to share and discuss their ideas and perhaps to adjust directions of their research, to set new acquaintance in order to start new collaborations. This could be the main arguments why we should return to the classical physical format if circumstances permit. One could also argue that classical conferences are the thing of the past, that they are just a form of academic tourism, and that nowadays no one really has the time and motivation to attend conferences in physical form. Certainly, there is some truth to such reasoning, and online tools have their place, but evolution did not prepare us humans for interactions via digital interfaces only. We are still social beings. For the newcomers to the conference, some short historical facts. So the CESP conference series evolved from the Alpe Adria scientific collaborations and was launched in 1995 under auspices of OIFEPS, with the goal of creating a common platform for colleagues from the Central European region working in the field of pharmaceutical technology in industry, universities, and institutes. The symposium nowadays traditionally gathers colleagues from all over Europe, and in addition to the goals of a classical conference of a bigger format, offers a good opportunity to promote scientific achievements of talented young pharmaceutical scientists. I would like to thank the heads of pharmaceutical technology, biopharmacy departments, and the heads of the research and development from the pharmaceutical industry across Europe for their past and current support to the conference series. So 13th Central European Symposium on Pharmaceutical Technology, entitled Contemporary Pharmaceutical Technology, Addressing Challenges of Innovative and Generic Medicinal Products, is addressing 4 key topics. It is of formulation concepts and delivery strategies to improve patients' compliance, of continuous and additive manufacturing of drug delivery systems, of approaches to improve stability and bioavailability of biopharmaceuticals, and topics in compendial and biorelevant dissolution testings. Participants will also address how pharmaceutical technology provides tools to combat COVID-19 pandemic. At the end, I wish all participants a fruitful conference and enjoyable experience. Thank you for your attention.
Thank you, Rok. Closing this welcome short session, I would like to present some numbers. I am pleased that we have participants registered in the number of 159. Even during last day, 10 more registered, so we are very pleased. They come from 18 you come from 18 countries. The program contains 12 plenary and keynote lectures and 9 short communications. Not everyone, of course, could come to Gdańsk due to still some health risk. Thus, 10 speakers will present the pre-recorded lectures. In the continuous poster session, 75 posters will be available for online presentation and discussion, while 9 posters will be also presented as short videos. Now I would like to ask Bartek Maciejewski, the secretary of our symposium, who will present some of the information, technical information about these internet interactions during the symposium.
Ladies and gentlemen, a few words about technical matters regarding your participation in this symposium. First of all, oral sessions. If you would like to ask a question or add a comment to the presentation you are attending, if you are present here at the lecture hall, just raise your hand and our staff will provide you with a microphone, and then you will have the opportunity to ask your question. If you are present online, there are 2 ways to ask a question. First is asking your question by typing it in the chat box, and our staff will provide it to the speaker, who will try to answer your question. The second possibility is to click raise hand or type I have a question or some similar phrase in the chat box, and you will be allowed to ask question by yourself. As for a poster session takes place continuously during whole time of the conference under the address provided on the screen. And additionally, 2 screens for poster viewing are available in the lobby for the participants that are present in the venue. For using the online poster session, you need to log in to the forum by clicking the button login and then providing your credentials, which have been provided to you via email. After logging in, you will see two categories of posts, oral presentations and posters. The category oral presentations contains all abstracts of lectures and presentations that will be presented during the conference. In the section posters, you will see abstracts and full posters that have been provided by participants. After clicking posters, you will see titles. After clicking on the title that you are interested in, you will see a short abstract, and by scrolling down, you will get to the actual poster. If you want to comment the poster or start a discussion under the poster, just click post reply, which is on the at the bottom of the page, and afterwards you will be able to type your message, which will be provided to the author of the poster by sending him a notification. If you are particularly interested in some posters, you can subscribe the topic, and you also will be added to the list of participants which receive notifications when something happened below the poster, meaning someone asked a question, someone answered the question, or there is some ongoing discussion. We would like to ask you to check the comment section, to log in to the forum and check the comment section on regular basis during the symposium, and answer the submitted questions as soon as possible, so that we can take as much of a discussion space as possible. And if you have any questions or technical problems, you can send an email to the address provided on the screen, or just ask someone with yellow badge. Thank you very much.
So now is time to start after this starting session. And we are very happy that one of the godfathers, as I mentioned, of this CESP series of events is with us in Gdańsk. This is Professor Douwe Breimer from the university, from Leiden University, from Leiden Academic Centre for Drug Research. Professor Douwe Breimer, since 1975 is a professor of pharmacology at this university, and what is very also important, he was a rector of the university and president of the executive board in Leiden University. He holds honorary doctorates from several universities in the world. His research focuses on pharmacokinetics, pharmacodynamics, and drug metabolism. The citation number is really marvelous. These are almost 10,000 of citations of his research. So, Professor Breimer, we are very happy that you are with us, and we are looking forward to listen to your thoughts, opinion, how to improve patients' compliance by combination of drug therapy. With all your knowledge and experience in pharmacology, and also in connection with drug formulation. So please, Professor Breimer, the podium is yours.
Thank you very much, Professor Snitowska, for this very kind introduction. And it indeed gives me great pleasure to be physically present after such a long time of only staying at home or in the laboratory. It is now possible again, at least to a certain extent, and for limited numbers of people, I have to admit, to be present and to meet again. And as some of the introducers already said, we are here at the university to meet preferably face-to-face, because that's basically what education is preferably all about. But I have to admit that a lot of innovation has been developed in recent months, in recent years, almost 1.5 years, to actually also develop technology that makes interaction online very much possible. So the hybrid event is somewhat of an intermediate advantage, I would say. I would like to have my first slide, please. This is not my first slide. As Professor Snitovska already said, I'm basically a pharmacologist, but trained as a pharmacist, and always have had a keen interest in pharmacokinetics, pharmacodynamics, and the relationship with drug delivery, with pharmaceutical technologies. Some of you have heard me in the past also talking about pharmacodynamics of drug delivery. And in recent years, I have developed a keen interest in systems biology, systems therapeutics, and the challenges for drug delivery, as I've indicated as the title on this particular slide. You may remember once upon a time, and actually not even too long ago, medical doctors used their freedom to prescription abundantly, while pharmacists compounded extempore almost every drug combination possible into pills, powders, potions, and capsules. And pharmacist, I was trained as a pharmacist, as I said, we actually made that all possible. Which was relatively difficult sometimes, a challenge, but we did it. But so combination therapy was more the rule than the exception. However, the combination per se was usually not based on proven clinical evidence, but was just based on empirical evidence, some experience by medical doctors, and of course published in the literature as well, but it was not real clinical evidence. And that led to having combination therapy actually somewhat of a bad reputation, namely polypharmacy. So if you have one drug, it gives of course the potentially beneficial effect, but potentially also the adverse effect, leading to drug number 2, also having an adverse effect, leading to drug number 3, and that gives rise to, as I said, polypharmacy, which is now very much frowned upon, as I said, a bad reputation. For example, a typical rheumatoid arthritis patient, which I took from Bert Leufken's presentation in 2007, 2 to 5 prescribers, 2 NSAIDs, TNF blockers, oral steroids, osteoporotic drugs, cardiovascular drugs, etc., etc. So one drug leads to the other, which of course is not the desirable type of therapy that is to be pursued. Potential risks and disadvantages, let me start with that. Adverse effects, polypharmacy, as I just said, drug-drug interactions, unwanted, negative influence on adherence to the prescribed regimen, patient compliance not as desired, and most often, as I already said, not evidence-based, no distinction possible between the contribution of each individual drug to the main effects and adverse effects. So it was relatively confusing which drug caused what. And the potential drawbacks of fixed dose combination, FDCs, are adjustments for individual patients is more complicated. Addition of the different adverse reactions specific to each substances cannot be entirely identified. Drug-compound interactions in a specific formulation. Of course, we knew that as pharmacists in the old days when we had to compound or to actually make all those combinations. And bioequivalence assessment, of course, as some of you may be aware of, is very much more complicated. In drug regulation, the EMEA guideline on fixed combination of medicinal products, there is a reluctance, namely only if potential advantages are based on valid therapeutic principles and substantiated by clinical evidence, it can be accepted. And that means that addition or potentiation of the therapeutic activities of the substances used alone at higher doses than in combination. So the 1 + 1 is bigger than 2. That's what we call in pharmacology if there is synergy, or is associated with a better safety profile, so that 1 + 1 is smaller than 2, than the added products, then it may be acceptable. Or it may be acceptable if the counteractivity by one substance of an adverse reaction, serious or commonly occurring, is produced by another one. So then also a combination may be accepted. I'll give you some example later. Or a simplification of therapy by decreasing the number of individual dose units to be taken by the patients, which simplifies therapy and may improve patient compliance, which is also a very important aspect of combination drugs. But I will refer to the polypill later in my presentation as an example of this C. Examples of, so there are quite a number of fixed dose combinations have been registered over the years, which have made sense, and I'll explain why they make sense based on this synergy in particular. An ACE inhibitor, for example, is antihypertensive. An ACE inhibitor with a diuretic, a beta blocker with a diuretic, or an alpha blocker with a diuretic, calcium channel blocker with an ACE inhibitor. So there are quite a number of combinations possible and desirable under certain conditions for antihypertensive. Cardiovascular, antihypertensive with statins. Of course, they have not the same mechanism of action, but they are very often combined in cardiovascular disease. And of course, that has led to the development of the polypill, antihypertensive, acetyl salicylic acid, and the statin, all combined in one pill, which increases indeed adherence to that particular combination. And in particular, polypill has drawn a lot of attention over the years. It was published initially in the Lancet on on a study in India, and has been followed up by very many countries in this particular example. But also in diabetics, it is not uncommon to have good fixed drug combinations. Or antivirals, HIV. You can hardly think of HIV treatment without a cocktail of different drugs, of antimicrobials, cotrimoxazole, and you may know, those of you aware of therapeutics, or opioids, the combination of oxycodone and naloxone, where the naloxone counteracts the side effects of the oxycodone. So the FDA actually lists approved FDCs, fixed dose combination, since 1961, and I was struck by the high number, more than 1,300. So although there is a reluctance among the regulatory agencies to actually approve these type of combinations, there are over the years many have actually been approved for the reasons which I just indicated. And I'll now move on to the reasons why this actually makes sense and actually also represents the future, in my view, to a certain extent, of drug therapy. So what is the rationale? And that is derived basically from the regulatory requirements published by the FDA as well as by the EMEA. Additive or synergistic effects, that's what we should like to pursue, affecting the same pharmacological endpoint or clinical endpoints through different mechanisms. And I showed you the combination or the examples: antihypertensive combinations, antiviral combinations, anti-diabetic combinations. There are quite more. Or complementary effects affecting different pharmacodynamic endpoints to reduce health risks. So that's where you have the example of antihypertensive agent with a statin. Or cooperative effects affecting different pharmacological mechanisms, leading to reduced resistance and enhanced activity or reduced adverse effects. So for example, antibiotics, amoxicillin with clavulanic acid, or opioids, as I indicated, oxycodone with the antagonist naloxone, which then has the action peripherally. Or convenience and enhanced patient compliance, and what's I indicated before, an example of that is indeed the polypill. Now, what is the actual pharmacological background of this? If you look, for example, into hypertension, this is one of the pictures taken from the famous Muchler's textbook on drug actions, where it is indicated that blood pressure is indeed regulated by different organs. So you see them on the slide, the vascular wall, the kidneys, the brain, the heart, and maybe some other mechanisms as well. So in other words, blood pressure is not just one mechanism, but there are several mechanisms of organs involved in controlling blood pressure. It is not a mechanism that stands alone. So I indicated on the previous slide, let me say, the different organs that are involved, but of course there are interactions between those organs, which we have to keep in mind. The body is, let me say, in a steady state, more or less, with respect to hypertension, and what means that if you indicate or you interfere with one mechanism at one organ, it may well be that the other organ or the other mechanism counteracts it. So in order to achieve, let me say, the desirable effect, you may have to combine different mechanisms of action, and thereby, of course, different drugs at the same time, to make sure that the effect caused by the initial, for example, therapy, is not counteracted by another organ through a different mechanism. So there is this interaction between the different organs and mechanisms involved in regulating blood pressure. And that gives rise to the need for interfering at different targets at the same time, as I will indicate. This is somewhat of a difficult slide to read, but it is basically the different mechanisms by which viral replication takes place in HIV. And also there, it is very essential that you address and use those different targets simultaneously at the same time, so that there is no escape anymore for the virus to actually replicate. So you have to address all the mechanisms at the same time. And that is the reason why HIV is to be treated by a cocktail of different drugs affecting different targets simultaneously. So here it is essential that different targets are treated at the same time. This is again a somewhat complicated slide, but it basically shows you the different mechanisms in cancer and cancer growth. Of course, you all know that there are lots of mechanisms by which cancer can grow and also mutate and replicate, or whatever mechanism you may call it. And that is also the reason why we have a whole arsenal of anti-cancer agents in actual therapeutic practice. But also there you see more and more anti-cancer drugs being applied simultaneously. So not just one anti-cancer drug for a patient, but more than one at the same time. And that makes, in my view, a lot of sense to try to avoid that indeed the cancer grows through a different mechanism than the original cancer treatment intended to. So that leads me to, and that, as I said, that has stirred my interest in recent years when I chaired a national committee in the Netherlands on the future of systems biology. And system biology can be defined as the quantitative analysis of the dynamic interactions between several components of a biological system, and aims to understand the behavior of the system as a whole, as opposed to the behavior of the individual constituents. And that is basically away from reductionism, so understand all types of single mechanisms, but trying to understand the integrated system when all those separate mechanisms work together or interacting. There's a wonderful little booklet by Professor Noble from Oxford that is called The Music of Life. And that's where he explains that although you may have an understanding of how the heart beats in terms of the different mechanisms which have all been studied in vitro, etc., but by adding all those mechanisms, you still do not understand why the heart beats. You can only understand that if you study it from the other end, namely study it in vivo. So the in vivo studies remain to be extremely important to understand how a system works, and that's the reason why systems biology is such an important development on top of, let me say, all the other types of experimentation. And that's the reason why I think that systems pharmacology is very important to understand drug action in the future and identifying new drug targets. So systems biology, so you see here on this slide the interaction with different molecular targets. You see the translation into a network of genes, biochemical cells, tissues, organs, and that leads to an endpoint, which is not just one straightforward mechanisms, but it is based on an interaction between different mechanisms in an in vivo system. So that is why we have to study the system per se to understand what the outcome will be and how to interfere with it. So medicine's discovery, design, and development is to be based on the biological network model. It explains the resilience of a biological system with respect to drug action and disease processes. Resilience means how sturdy is it to resist to certain changes, and how can we actually overcome these. And you have to therefore understand drug action as well as the disease, which I think for pharmaceutical science is very important as well. Identification of novel pathways of diseases. So that's what you have to understand. Let me say what pathways are involved in a disease, and thereby identification of novel drug targets. And then the design of novel therapeutic interventions. So that's basically what systems biology or systems pharmacology is all about. And that ultimately leads to what I call systems therapeutics. This is a very complicated slide again, but this is only to show, for example, what one understands in osteoporosis. This is a whole complex of disease mechanisms. And in red, you see the interventions with drugs, and also showing that a combination of drugs is probably more suitable for the actual treatment than single drugs. But understanding the mechanism of the disease is extremely important. But coming back to our own science, pharmaceutical sciences, systems biology of disease, defining new drug targets, leading to systems pharmacology. Then you have to study systems pharmacokinetics, pharmacologically based PK. I think we have one of the lectures later this afternoon on that particular topic. Systems pharmacodynamics, studying the drug interactions. PK/PD as an essential component, as I will show you, and then ultimately lead to system therapeutics. And the system therapeutics may involve multi-target drugs of rational single-target drug combinations. So the combination of drugs on different targets. And remind you, I have put here multi-target drugs. We have for a long time believed that a drug should be as purely as possible, as selective as possible on one target only. And if it were not pure or selective on one target, we called it a dirty drug. Now we call it a multi-target drug. Now all of a sudden that has become a very desirable property. So also in the industry, people are looking if you can combine in one molecule, in one drug molecule, interaction with different targets simultaneously to actually achieve synergy. That is not so easy to have to combine from a medicinal chemical point of view, to combine those properties into one molecule. And therefore, the escape basically is to actually use different drugs which have ultimately the same effect, but in combination so that synergy is actually being achieved. So what you need for that is pharmacodynamic drug interaction mechanisms. I only will discuss these two receptor systems. So you have two receptors on different organs, for example. Think of blood pressure, drug A and blood B, and drug B. And they both lead to an effect on blood pressure, so E, but there is an interaction between the two. And that's where you need pharmacokinetic, pharmacodynamic modeling to actually see which ratio of those two drugs give rise to that synergy. So this is the two receptor model, A and B, drug A and drug B, and leading to the effect E, which is caused by the two drugs, but what you want to achieve is actually the synergy between the two. And that requires PKPD modeling. This is again a somewhat complicated slide, but some of you who understand PKPD will recognize it, that you have a three-dimensional picture where you have on one axis the concentration of one drug, on the other axis concentration of the other one, and then the difference in effect is then on the y-axis, and then the red area, so you look actually at the surface, the red area where the synergy is being achieved. In other words, you need a certain concentration ratio where that synergy is being achieved, and that requires sophisticated PK/PD modeling. Now, how does that relate to drug delivery? Because that's what I ultimately was going to discuss in this lecture very briefly. What you want to achieve is actually a constant ratio between those two drugs. But in actual fact, if you have two compounds, A and B, there is always differences in pharmacokinetics between the two. So A may be eliminated very rapidly, as you see here, single dose, and B may be eliminated more slowly. Whereas in actual fact, if you would give them in a fixed dose combination, you would have very fluctuating concentration with drug A and more steady concentration with B. Whereas in order to achieve synergy, you have to achieve more or less a constant ratio between A and B. Now, how do you achieve that more or less constant ratio between A and B? That is the challenge, in my view, for drug delivery, namely, meaning that you need a combination, a drug delivery system from which A and B are delivered at different rates. So you have a delivery profile which is different for the two different compounds, for A and for B, to achieve a more or less constant ratio between A and B. Is that technology available? Yes, I've always been impressed by the work of Professor Paolo Colombo in the University of Parma with his dome matrix technology, where you can actually put different compounds in different parts of the matrix and then see what different release profiles you can achieve. I don't give examples, there are lots of examples in the literature, but the technology is there to actually achieve this particular objective with this type of technology. And the other technology which I think is very promising is, and I know that is also a topic at this conference, and that is 3D printing. So 3D printing of dosage form, I think, is a wonderful tool for the future to achieve the objective that I just described. There's a lot of literature already available. It is still in its infancy, I would say, to actually use it at larger scale. but it is very promising for achieving what I believe the future of drug combination therapy, systems therapeutics, to achieve that through 3D printing, where the different layers of your 3D printing product will have different release profiles based on, let me say, the composition of the different matrices of that particular product that is coming out of the printer. So I look forward to seeing a lot of those type of results, and that leads me to my basically wrap up, namely prediction of pharmacology in man in terms of multi-target treatments. Therapeutic aim is synergy, enhanced therapeutic efficacy, design and development, selection of a combination of drug targets, as I indicated, derived from a better understanding of disease mechanism, selection of a combination of drug molecules, prediction of the combined drug effects through PK/PD modeling. So here again, pharmaceutical sciences is key for such developments. And I should be a key component of pharmacy education, in my view. And then, of course, the selection of the appropriate drug delivery system. So in other words, pharmacology cannot achieve this without drug delivery innovation. And so that's the reason why I think that systems therapeutics requires innovation in drug delivery, difference in the delivery rate of each separate compound needed based on PKD data, and flexibility needed to cope also with intersubject variability, and also there 3D printing could in the future be of great help. So this is what I would like to leave with you as a final message, as a challenge for those in pharmaceutical technology who are interested in innovation in drug delivery, and of course engage in multidisciplinary efforts to achieve what I call the future of therapeutics, which is systems therapeutics. Thank you for your attention.
Thank you very much, Professor Breimer, for this inspiring lecture, and I believe that it's not only on the topic which is actual at the moment, but will be really actual for the next many years. The lecture is open for discussion. Maybe there are some questions. Yes, Professor Polak.
Thank you very much. Great lecture. I have a very practical question. I'm sure you know this term virtual twin, which kind of is in parallel with the topic of your lecture. How much time do you think we need to make it practically available in the doctor's prescribing or doctor's room, prescribing the drugs to the patients to really deal with the virtual patients or virtual twins, and based on the modeling and simulation, prescribe the drugs to those patients?
Yes.
When will it happen?
Well, it's an interesting question. I cannot predict the future, but I think it always starts with, let me say, putting a lot of effort in the research in order to achieve this ultimate aim. And I think that what I see now, for example, in the Netherlands, what happens in hospital pharmacy, for example, they are already engaging in 3D printing, etc., and delivering combinations which so far were relatively difficult to deliver. And of course, it's still on a relatively small scale, individual basis. But since we all aim at precision medicine and everything associated, this should be taken as part of precision medicine, individualized medicine, etc. It's a long way, I have to admit. I gave my inaugural lecture in 1976 called Tailored Pharmacotherapy, so individualized pharmacotherapy, because I was aware of the huge inter-individual differences, but I knew at that time hardly anything about the mechanisms. Now we understand far more about the mechanisms, and taking those into account, we are also basically able to ultimately achieve a far better result than we have so far. So this is I am aware this is still somewhat of a vague answer, but we will get there. We will be getting there. And you are still young, so you will still experience it.
Maybe more questions? Yes, Professor Tuleu.
Thank you. I have a question. So you set an example, you show this matrix dome technology, it's combining different components into one pillow or tablet essentially. So do you think that 4-component systems, that like mini tablets filled into the capsule with 4 components, is it solvable for many systems that's combined 2 drugs? So to solve this problem of having, compensating different elimination rates for 2 drugs with 4 components, because this is why I'm asking, because this is industrially achievable technology.
I cannot answer your question very specifically because that's the reason why I have a lack of, let me say, pharmaceutical technology and knowledge to actually answer that question. But I would say in principle, if I see, let me say, what all those different matrices, etc., are able to achieve, in principle, that should be possible. Of course, it depends also the loading dose, and then of course on the type of matrices which have to be combined. There may be also chemical incompatibilities, etc. But in principle, that should be achievable. Yeah.
Thank you very much. We'll continue this discussion in the break, probably, but we must continue. Thank you, Professor Breimer.
My pleasure. Thank you.
Our next lecturer will be Professor Catherine Tuleu from the University College London School of Pharmacy. Professor Tuleu is a professor in pediatric pharmaceutics since around 2003. Her main focus became research concerning drug delivery systems for children, including neonates, infants. This research is linked to the various routes of administration and very much to acceptability of dosage forms and patients' compliance. Professor Tuleu works closely with pediatric hospitals and pharmaceutical companies. Part of Dr. Tuleu's work is also to ensure that those pediatric formulations are, of course, safe. Very important is that Professor Tuleu is a founder and chair of the European Pediatric Formulation Initiative Consortium. By the way, next week there will be an annual conference of this consortium. Very important also from the point of view of people working on pharmaceutical technology. Professor Tuleu's Professor Tuleu's research is very highly cited, more than 2,000 citations. This is really a sign how important is the work Professor Tuleu is doing. The title of the presentation, which is first presentation in our plenary session, formulation, concept and delivery strategies for improving patients' compliance. This title of the Professor Tuleu's lecture is Kids Compliance and Beyond, a Matter of Taste, which is also the most important topic in medicines for children, which are not only tablets, but also liquids, for example, that the taste is so important. We have the lecture pre-recorded, and after the lecture, we'll have opportunity to speak to Professor Tuleu online. So please, we will start with the lecture.
Hello and welcome to my remote lecture on Kids Compliance and Beyond, a Matter of Taste. My name is Catherine Tuleu. I'm Professor of Pediatric Pharmaceutics at the UCL School of Pharmacy, London, and I'm also the founder and the chair of the European Pediatric Formulation Initiative. I felt I had to mention it early on, as I'm going to use some of the work that has been done by this group, this consortium. So I've been tasked to talk to you about medicines for children. So my storyline is that I'm going to try to tell you what was or is happening in practice with medicines for children, what is or should happen in terms of pediatric drug development, and link that to formulation design, good formulation design. And we'll discuss as well issues around compliance and acceptability, with emphasis on palatability, because this is an area of research I'm very fond of. So to set the scene, so what's happening in practice? Well, if a medicine is licensed for pediatric use, meaning that in the summary of product characteristics, the indication and the age group is covered, and if the product, the dosage form, is appropriate for the age of the children, which is not always the case, then the commercial preparation can be used, and this is what we call license used. However, there are some other instances where the medicine is not licensed for pediatric use, yet is used. For example, atenolol syrup is used in children despite being licensed for adults. Then it's called off-label use. And then there is the gray area in the middle, where if there is no age-appropriate dosage form available, and the medicine, whether it's licensed or not, needs to be reformulated from raw material or manipulation of the adult dosage form, then it falls under unlicensed used. There's various ways it can happen. If it's reformulation, it can be done under GMP. In UK, we've got special manufacturers that do that, or it's done at a pharmacy. And then it's called compounding or extemporaneous preparation. Another unlicensed use is importation of dosage forms. It means that a product is available somewhere out of your jurisdiction in an age-appropriate dosage form, and that would be, despite some hurdles in cost and access, that would be possibly the best option. And of course, what's happening a lot is uncontrolled manipulation at the point of administration, where parents and carer have to open a capsule, crush a tablet, do all sorts of things to accommodate their children. So why is it a problem that that some unlicensed products are used? Well, for many of them, there is a lack of testing for purity, potency, content, stability. There is obviously limited stability data, if any. Often it leads to inaccurate dosing. Taste can become an issue because if a tablet has been developed to be swallowed whole, if it's suddenly crushed, it releases the taste of the API. It can alter the absorption of the drug, and there is obviously no or few bioavailability data on this manipulated or compounded formulation. It can lead to environmental issue, and in mind, if manipulation, for example, are done at home, that can expose the healthcare professional, or on the world, that can expose the healthcare professionals and or the parents to toxic APIs. And on top of that, there is a lack of awareness of what is a licensed product by physician, and there is no or very weak oversight by regulatory agency. I've put this graph from a recent paper that I've co-authored with my colleagues from a formulation expert group on Connect for Children network. And trying to see if progress have been made, we looked at the number of publications on compounded formulation for children of any age. And as you can see, in between 2000 and 2020, it doesn't seem to decrease at all. And we are in a paradoxical situation because depending where you start, let's start by, there is the drug that the doctor prescribed. This is my blue box here. Then of course, there is what the industry has made available. Then it goes to the pharmacy, and depending on what is available, there is what is dispensed. And then there is what's happening at the patient level, how it's taken. Is there any manipulation that is happening? Is it mixed with food, etc., etc.? So really, we are at the risk that from for example, an intact tablet developed with an optimized PK profile, we're completely changing this absorption profile, and that can lead to being in subtherapeutic or sub-optimal dose. And I think the reflection, on this situation is that, in fact, at any point, the children have been asked what they would be able and willing to take to input into this circle. And as an example, I used one of the early work I've been presented with. I received a phone call from a large pediatric hospital in London, and in a baby that had a nasogastric tube, they found some black seeds in the stomach aspirate, and they thought it was a case of poisoning. However, they knew that the baby was receiving the MUPs, low-sec omeprazole MUPs, so dispersible tablets, 10 milligram dispersed in 10 milliliter, and an aliquot of 3 milligram was administered through a nasogastric tube, a very thin one, 6F, and that was for gastroesophageal reflux on a patient on pediatric intensive care unit. So they asked us to look into that. So we got some samples, and in fact, looking at those beads that we found, we worked out that they were the remaining of the polymer, of the enteric coated polymer of those MUPs, small particles, tainted with the product of degradation of omeprazole, which is deep purple. And really, the realization was that if it degrades in the stomach, we have no idea of the dose that has actually reached the duodenum and has been absorbed. So we've got no idea what we're giving to these patients, whether it helps with gastroesophageal reflux or not. And the second realization we had is that it's all nice and well to develop a product like that, but in fact, in children, and especially in children under 1, there is a loss of variation in terms of gastrointestinal transit and other physiological values, and therefore we've got no idea how the small bits transit. And this was done kind of like more than 10 years ago, and these statements are still valid, I guess. Another recent project I've been asked to work on is the repurposing reformulation of pyridoxal 5-phosphate, PLP, which is used in a rare disease, epilepsy, for children who have a deficiency in pyridoxamine phosphate oxidase. And they are treated with PLP. PLP available on the market are dietary supplement with sub-optimal quality, and we've looked at that. So we've got no idea when they say 50 milligram on the label, most of them don't have 50 milligram in them. And as you can see, they come in a variety of type of dosage forms, tablet, capsule, which are rather large. And at the end of the day, this is the patients. So you've got screaming baby, you've got to crush the tablet, open the capsule, put it in some water, make as best as you can a dispersion, and try to withdraw a dose appropriate to the weight of the patient. We know as well that the compound is unstable under light, and it can be kept for quite a while in solution. And there is a risk that the degradation product could be liver toxic. However, there is also an unknown is that it could be the liver toxicity could be due to the dose given, because there is a need for a dose finding study, but without the right product, this is not feasible right now. And another important point is that this soup, this yellow soup, is not palatable and is gastric irritant. So really the idea was to try to reformulate this compound. But the situation should be better and is better since the introduction of pediatric regulation all over the world. I give the example here of the EU pediatric regulation. So since 2007, January 2007, pharmaceutical companies have an obligation to provide data in children, and this needs to be agreed as per a binding PIP, pediatric investigation plan. This is true for all new medicines, but as well new indication, new route of administration, and new pharmaceutical form. This PIP needs to be prepared by the end of phase 1 in adults. Of course, you can have a waiver if the indication doesn't occur in children, or a deferral if more adult studies are needed. What is important as well is that all pediatric subsets, so children of any age, need to be discussed. They might not all need it, but it needs to be discussed. The second pillar is that there is a pediatric committee that screens this pediatric investigation plan, and part of this pediatric committee, there is a dedicated formulation working group. This is recognizing that the issues around pediatric formulation in children might be quite unique. And there are rewards, incentives for pediatric studies that are conducted if the information is included in the SPC, and that is even if the clinical trial do not show efficacy. So I've just talked about the pediatric investigation plan in Europe, but there is a pediatric study plan in America, so under the FDA governance, and this is quite similar, and the two bodies, regulatory bodies, talk to each other, except that the pediatric study plan, the PSP, is needed in phase 2. But as you can see, I've highlighted on this slide that both in this pediatric plan needs to account for justification of the pediatric formulation development or an age-appropriate formulation development. And this is quite important as well. So, of course, the objectives are much broader in terms of, this pediatric drug development needs to reach an ideal best pharmaceutical for children. And of course, it's based on efficacy, it's based on safety, it's based on detailed dosage recommendation, and as well, a product that is accessible and is reasonably cost effective. But what I want to discuss more specifically is that there is a requirement as well for a suitable dosage form. So convenience, that leads convenient and accurate administration, a dosage form that is acceptable, palatable, with practical storage condition, and is portable. And really, that shouldn't impair the quality of life of patients. And one thing, I've highlighted these issues of cost and access because there are 2.2 billion children in the world. 90% of those are in developing countries. So we need to think of other settings where that might be condition might be very different. Condition of use might be very different than in our more privileged countries. So how can we develop children-centric dosage form? It's based, so those subsets I've just mentioned before, they are based on ICH topic E11, which is a guideline around clinical investigation of medicinal products in the pediatric population. And this guideline states 4 subsets. So there is preterm and term infants, so 0 to 28 days. Newborns. Then there is infants and toddlers from 28 days to 23 months, so up to 2 years of age. Then there is the children category, 2 to 11, and adolescents over 12. And this is mainly based on developmental age because this is to guide the clinical trials. And we know that there are differences. There are differences on transdermal drug delivery that affects transdermal drug delivery. There is differences in gastrointestinal functions. There is a higher respiratory rate. The kidney filtration might be very different, especially, and this is especially true for early, younger subsets of the pediatric formulation. I think in terms of formulation, so that would mainly affect PK/PD, I guess, or directly linked to PK/PD. But for what we're concerned in terms of formulation, it is important to consider the physical and behavioral age. And therefore, the categories of children 2 to 11 is a very broad category where many changes occur. And I think for us, it's easier to consider this category as 2 to 5, so preschoolers, and 6 to 11 school-age children. And that's all that to say that really we've got a moving target in terms of defining the product that is going to be the best for the patient population when this patient population changes that much in short amount of time. Well, I really like this cartoon because it's an easy way to capture attention on the various issues around giving medicines to children. The route administration can be quite tricky here. We've got an example of parents trying to give eye drops. We've got as well the rectal administration, which for various reasons, might not be that liked, I guess. The type of dosage form is also important, and the dosing. So for example, you can see here that despite having a liquid in a syringe, this baby is spitting out most of the dose, so we've got no idea what we're giving really. The use of administration aids is often happen often for children, and you can see the mom at the top who is trying to make the medicine go down with some chasers that the kids would like. And of course, main issue is the taste of medicine. And this is important to understand that probably as an adult, we are much more resilient when it comes to bad taste of medicines, but children think very differently. And this is what I'm going to try to focus on today. So there are obviously many factors that can affect adherence in children, and this is not the remit of my talk today. But I think what I want to emphasize is that for children, maybe medication factors are upfront factors that are major challenges. And there is this recent research paper published in 2019 that asked many European children, why do you find some of the medicines difficult to take? And as you can see, 652 answered. And clearly above any of the answers, it's because they don't like the taste of the medicine. And it's above it hurts to take the medicine. So things like an injection or, unpleasant feeling, etc., etc. So clearly, this is the number one issue we have to deal with. And I noticed that the journal that published this paper shows a picture of a little girl, I think, refusing to take her medicines because of the taste, possibly. So linked to that now, we've started in the field of pediatric formulation, we've started to talk about acceptability. So there is an official definition, and it's the overall ability and willingness of the patient and the caregiver. And that's quite important because obviously there are some children that are completely dependent on their caregiver, whether it's a family member or a healthcare professional, and that's defined as user to use a medicinal product as intended, as authorized, so as it's described on the summary of product characteristic. I think it's important here to remind ourselves that what we call medicine is the therapeutic entity delivered to the user. So it encompasses the type of dosage form, so the formulation, but as well the appearance, the tablet, the size, the shape, the color, the dose of the API, the dosing frequency, the primary packaging, if there is a device associated with the administration, the container closure system, and the written instruction. And I'll illustrate that later. And why? So then this ability and willingness. And here you've got the example of, this kid being given like huge capsule, and the pharmacist or doctor commenting, I hope you're not one of those kids who have trouble swallowing pills. And so that's more around the ability, and then, or maybe the willingness, but clearly picture two, you've got this child who really doesn't like the taste of our medicine, is refusing to take the medicine. So that is compromising the PK/PD and is really, a step back in terms of compliance. So what are the acceptability considerations as per the official definition? Well, there is, to start with, it's what you see. So that's going to be the color, the shape, the embossing, etc., that the dosage form shows. Then the second thing is what you smell. And once you've put it in your mouth, is the taste, the mouthfeel, and the aftertaste. So all those palatability aspects are extremely important. This is the first contact with the medicine, on top of having to take it out of the box. And of course, then after there is the swallowability. So it's obvious to think that if a tablet's got a weird shape and is really big, some children, as well as some adults, because we know that probably a fifth of the population is struggling with swallowing tablets and capsules, but it's even more acute in children when they're presented with a rather large dosage form. But as well for liquids, the volumes that you deliver is very important to consider. And that's, of course, a bit linked with the required dose, I guess. Importantly, you need to consider how frequently and for how long you need to take the medicine. So probably acceptability issues are going to be different when it's for an acute or a chronic condition. And an important aspect is in between the moment you get the medicine out of the box and you're ready to give it to the children, what are the different steps, how easy it is. And that's all this complexity of modification per administration. If there is a use of an administration device, how easy it is, as I was explaining, the primary and secondary container closure system, and as well the actual mode of administration. And I gave this example. I'm not going to go in details, but this is really counterintuitive here because, there is all sorts of kind of for a lay member, a parent, maybe in a resource poor setting, you need to check if there is some bubbles in the syringe at 2 stages. You need to swirl, but don't shake. That could be quite difficult. You need clean water. In some settings, you don't have necessarily access to clean water. And this product comes with 3 different syringes. So again, in terms of literacy skills needed, you can see that it's quite complex. So this is, and this is for a product that has been specifically developed for children. Yet, it's still quite complex in terms of administration. So that was more around the patient consideration, but I think we need to understand that compared to adult development, acceptability is a pediatric extra regulatory challenges. Now it's formally defined in the latest revision of the ICH E11. And as well, it's important to note that it's a key binding element in PIPs. So if the pediatric committee as asked to look at acceptability, this is going to be a key binding element. For more information, the Global Accelerator for Pediatric Formulation, GAP-F, has published various modules, and module 5 is on acceptability. So if you want a full run through acceptability, that would be a good resource. Okay, so medicines for children should be age and ability appropriate, but also acceptable. So it's important to define your target product profile from the outset. And there is various ways, depending what's the key component. There is various ways to build this target product profile, but I like, this is a paper we published from the taste assessment and masking team from EUPFI, and this was our proposed approach. So as you can see, it's mainly based, there is the dose is important. How flexible do you need the dose to be? Is it per kilogram of body weight, or just a couple of strengths that can be dose banded for wider weight and age of children? But what is important is that very early on in this logical decision tree, the taste comes up. So your decision on the final dosage form can be based on how bad is the taste. And then, of course, in this one, we've tried to group formulation that would be more suitable for neonates, so formulation that would be more suitable for children 1 to 5 and children 2 to 5. But just to show that, so quite early on in your development, you might have to make a decision based on the taste. And of course, we've got various taste masking options available. Again, this is a paper from our taste UPFI workstream. So there is a little bit of traction at the moment on looking at bitter blockers. So something that would literally antagonize or block the receptors for bitterness. So that is, and that would be the ultimate goal, to reach a neutral formulation. There is lots of other consideration to do, but that would be the goal. Then of course, the other taste masking option that you might be more familiar with is obscuration of taste, where by changing the viscosity by using sweeteners or flavoring agents, you kind of fool the taste buds. You give a lot of sweet or other sensory triggers to try to deter from the bitter taste of the drug. However, if the drug is very bitter, and many are, this is something that is very difficult to achieve, to have enough taste masking that way. And that's as well what people use with food when you try to hide the drug in food. Another approach, it's a modification of the API, and that can be done quite early on. So changing the salt, changing the pH, so in terms of solubility, or using a prodrug. Then after, we are moving on to trying to cover this API, whether it's at molecular level or whether it's at dosage form level. So the first approach is creating a molecular barrier. So it's complexation, so using ion exchange resins or cyclodextrins. And the second one is literally physically coating a dosage form. And those two options are more appropriate for solid dosage forms, but again, then limitation on age range and how they can be used. But I think my point was to say, you know, we've got various options and that are more or less better suited for different type of dosage forms. But it's all based on how can we measure this taste then, and when can you measure this taste? So if I start at the bottom of my slide, literally, you know, what you do in clinical development, during clinical development phase, during the later phase of development, of course, there will always be a check in patient, or there should always be a check in patient, because, and even though it's not real life, it's part of a clinical trial. So ideally, post-marketing would be better. But if you do it at this stage, and you can sometimes use healthy volunteers, but that is, I mean, if it's safe to do so and ethical to do so, but at this stage, in any case, isn't it going to be too late to reformulate from scratch? So really, what is would be very valuable is to have method that could be used during preclinical development. So we've published a paper a few years ago where we're trying, in a simple way, we looked at various methods available, and we used at their usability, speed, ability to screen pure API or formulation, evidence of correlation with human taste data, potential for validation, and cost. That was kind of like an initial screening program, and we looked at non-human tools for evaluation of bitter taste. And what came out of that was the rat brief access taste aversion model, which I'll describe a little bit later, and electronic taste sensors, or e-tongue. But that's more or less if they are validated with human taste panel data, because, and those two models are mainly for, especially the Itong, for taste only, so really bitterness. Whereas, of course, humans can give you a full profiling of your drug, what it smells, what it feels in the mouth, etc., etc. And important to note as well, there is a way to use healthy volunteers during preclinical phases, but this needs to be, again, in terms of safety, ethical issues, this needs to be, and in terms of legal aspect, this needs to be appropriate to do so. Now, recently, we've just published, in fact, a paper in September. It's in press in JPharmSci. And this was a refinement of those methods to compare, to measure the taste of pharmaceutical early on. And the reason is that now we've, there is more and more method available, but because they are not used routinely, there is still a lot of not misunderstanding, but lack of understanding of what they can do and for what purpose. So I think this new assessment was a larger collaboration with colleagues at EUPFI, so a lot from industry, and try to look at applicability of the method described early on. And the scoring was much more refined, because as you can see, there was like 13 criteria on how to rate those various methods, if to measure API alone, taste of API alone, taste of formulation, and the stability of taste over time. I think one thing to note is that whatever you do, humans are still the best. And then depending on the application, the applicability of the various tools varies, and some are quite early on in development. But I'm sure that years to come will make that landscape change. And this tool is interactive, so you can assess for your own needs, and it's going to give you where you are on this red, orange, green scale, and which method to use. I'll help you to decide which would be the best method to use to measure your taste. Now, as a personal example, so taking example in my own research, this is what we do at the UCL School of Pharmacy. We have an instant electronic tongue. My colleague, Professor Duncan Craig, has one. I've worked for many years on the rat brief access taste aversion model, which I'm going to describe. We have the capability of doing human panels, swirl and spit type of panel in UK. This is not considered as a clinical trial, so we are allowed to do so. And I've developed as well a mini flow-through bioerven buccal system. And I think the idea is to use a combination of those methods, depending on the needs, depending on the type of the formulation. If it's a solid formulation, we might use this biorelevant dissolution model linked to either a human or a rat taste assessment. If possible, we can move on to further formulation screening with the electronic tongue. That's really to give you an idea. But I think for me, what is important is this access to in vivo data. So for many years, we've worked on this rat bata model. And I think the culmination was, the excellent correlation in between human and rats in terms of bitter taste. And that really shows you, out of what, 15 API, that we can really, we've built up a model to propose a classification to predict human taste response. So that's especially applicable when you've got very early on with very little tox data, enough to risk assess the work in the animal, but to really benchmark, if your API is going to be a real challenge or not, and what kind of taste masking technology you should start to consider. So really it's helpful to do taste profiling, assessing threshold that then can be used to measure taste masking efficiency in terms of bitterness release, especially if we are talking about a solid dosage form with our bio-relevant buccal model. It can orientate as well choice of salts and enantiomer. We've worked on taste of excipients as well, and to really understand better the capability of the model. Of course, we've done some work with juvenile and elderly rats to show if there was any benefits in using juvenile rats in terms of mimicking children. We've looked at the gender effects, and there was a stream of work on multiple sensory attributes such as bitterness, grittiness, and viscosity in the model. So it's in development, it has strong foundation, and there is still lots of work ongoing. So this is so lots of research, but as well, this is a service that we offer to industry. And in fact, the rat data model has now spin out, and I've just founded a spin out company called Simcyp Ltd, where we offer these services as well. Now, going back into the human panels, because this is ultimately the gold standards, and this is not as simple as it sounds, and there is numerous considerations to have. Of course, the study design is extremely important, and you really have to think on what is your main research question, and then think of on your study population, if it's done on healthy or sick patients, adults or kids, the number of panelists or volunteers or patients you need to use, the setting, is it going to be at home, in hospital, in school lab, is it a single or multiple exposure, is it swallowed or just swirl and spat, which can affect, you know, the result, and is it a standalone measure or is it a comparison? So there is a lot of consideration, and it's on top, again, I repeat, above safety, legal, ethical issues, and it is important to understand that those panels are logistically extremely challenging. Then, of course, the scale and the patient-reported outcome, or the patient-reported outcome you're going to use, becomes very important, along with the statistical method. And of course, the acceptance criteria you're using. And just in passing, again, as a EU PFI exercise, we are working on a guide to good pharmasensory analysis practice, because we feel that there is a real need out there. And I like this cartoon as well, because this is exactly the point I wanted to make. So there is a thin line between good taste and bad taste. Does anyone know where the line is? Well, in fact, you know, it's often considered that if the medicine is acceptable to 80% of the patient, it's okay. But we don't have any guidelines. And what about the 20 other percent? So I think it's got to be looked at, you know, within maybe the PK profile of the drug, the severity of the condition, etc., etc. So it's not that simple, and needs to be adapted to each situation. So I think to kind of close this part, early identification of key attributes to be considered for inclusion in a pediatric quality target product profile is very important. And we certainly have a need for harmonization and consensus on the assessment of acceptability, and that includes palatability, which is one dimension of acceptability. So going back to our liquids, because this is really what we know for children, you know, children will receive solution, a syrup, or so is it the gold standard? I want to use this example of the Caletra, so HIV treatment, lopinavir, ritonavir. So as a start, it's an extremely unpleasant tasting liquid. It requires refrigeration in practical, and it must be taken with food. So there's many limitations there, but especially in resource limited setting. And on top of that, and that's really a point I wanted to make about excipients, because this is another dimension of the pediatric product especially for younger pediatric subset that is very important to consider is the excipients. So this one contains 42.4% of alcohol plus 15.3% of propylene glycol, so another type of alcohol. And I've just shown you down below that whereas a beer contains 5%, a whiskey, a vodka, tequila will contain around 40% of alcohol. And this medicine for children probably contains more. So this is really important to consider. And are they really the gold standard on top of the multiple excipients needed in terms of quantity, quality, the importance, and again, I don't have time to talk about that, but there is various dosing device on the market and they're not all equal in terms of accuracy and precision. We know that in terms of, they might be more costly, maybe easier to develop, but could be more costly at the end, and they cost certainly more to be transported around the world. And they might be more challenging in setting in developing countries where most of the children are, as I explained early on. So yes, so are they the gold standard? But they might be the gold standard, but not the golden standard. So now solid is the new liquid. So I've just described the issues with the lopinavir-ritonavir liquid. We know that there's been some matrix that were developed, which were heat stable. They could be taken with or without food, so big advance compared to the liquid. However, you can't really see the scale here. They are huge tablets and they can't be chewed, they can't be broken, they can be crushed, they can be dissolved. So really limited to only the population of children that could do it or be trained to do it. And in fact, some children younger than our children would be trained to swallow that to avoid the liquid, because that was really that bad. However, in the meantime, on the same principle, some oral pellets have been developed, and therefore 3 months old and older or kids above 5 kilos. And there might be a multi-step preparation prior to administration, but it offers as well flexibility because the capsule, they can be taken all, or they can be put on porridge, on milk, on various hot or cold drink. This is probably breast milk. So really offer a bit more flexibility of administration, which is quite important, and have the same stability profile as the solid tablets. And that's a link to what is happening now in pediatric development in terms of trending. There is really a move towards, or an interest in dry and solid oral dosage forms. So has been developed and put on the market some powder for reconstitution. Again, they are called ready to use, but are they really, that's the example I gave you before, where there was like 12 steps to get to the final product to administer. And then, of course, since WHO campaign in 2008, where they started to introduce flexible solid oral dosage forms, we've seen a huge interest in, for example, dispersible tablets. And for example, there is a Puma. Puma is pediatric use marketing authorization. There are off-patent drugs that are reformulated in an age-appropriate formulation and can get a license. And this marketing authorization has a protection of 10 years for whoever is developing this pediatric product. For example, Quebec is vigabatrin dispersible soluble tablets that is licensed from 1 month to 7 years old. And it's a Puma. And then some interest in oral dispersible tablets, oral dispersible films, as you can see here, and multiparticulate. And that has been really a big push around that, where many people have undertaken some studies with placebo to show, to evidence the fact that they are safe and usable in children, even if very young. And the two main ones are pellets or granules and mini tablets. Mini tablet has raised a lot of interest. And as you can see, there is a Puma as well, melatonin 3 millimeter mini tablet, and hydrocortisone has been developed as well as a capsule for sprinkling on different strains as a puma. Maybe one point to consider, I won't have time to cover everything, but those solids, you might be able to mask the taste a bit better, especially for the multiparticulate one, but they bring new acceptability challenges, mouthful, mode of administration, compatibility with the dosing vehicle, etc., etc. And yeah, maybe like an hybrid version, which is quite interesting, the oral dispersible mini tablets, which offers flexibility of dose, provided the right packaging and all the right administration devices. Yeah, so an interesting evolving field with new products, solid for children, even very young. So I guess I want to stop, and if I've got one take-home message, is to try to make you remember that therapeutic success is obviously based on adherence, and I hope I've shown that in terms of medication factor, acceptability, especially for children, is a big one, is a big one to consider. And acceptability is not only a swallowability, it's palatability, which is highly depending on test. Hence the importance of this new field of sensory pharmaceutics, as I call it. It is important now we're gaining more and more knowledge on linking these aspects of taste and formulation and dosage form design factors and administration factors. This is important. So I would like to thank you for the organizers for the opportunity. And I wished I could have come to Poland, but it was not possible. I would like to thank you for listening, and I will be available for any questions. And I might take this opportunity for people who are interested. We're having our 2-day yearly conference coming up soon. It's the 13th conference of the EU PFI, and it's online. And it would be if you want more detailed information on the various fields around pediatric formulation, join us and join the movement. Thank you very much.
Thank you very much, Professor Tuleu, for this excellent lecture with many directions for us pharmaceutical technologists. And now the lecture is open for discussion, for questions. Maybe somebody from the audience or online. I hope that you are with us, Catherine.
I am. Can you hear me?
Wonderful. Yes.
Hi, everybody.
You. Yes.
Hello from sunny London.
Yes, we can see it. And I also admired your background during the lecture. It was a nice London view. Thank you for this. So what about questions? I will start maybe because you mentioned and you showed that there is one of the ideas is to make obscuration of the taste with viscosity agents, yes. And unfortunately, we can have also the problem with bioavailability when we introduce the micro pellets or mini tablets or suspension to the viscosity agent, but the same happens with the food actually. So what do you think about that? That of course we can very easily sometimes obscurate this taste, but what about the bioavailability, how it influences?
I think it would be dependent on your API, on the food sensitivity of your API itself, I guess. And obviously of the...
But viscous agents, viscosity, when you have viscosity in your formulation, like the gel, you may introduce suspension to the jelly food or to the gel to obscurate the taste. So finally, you may change pharmacokinetics.
Yeah, but that, you know, you would know quite a lot from your drug development in terms of food effect itself. So there would be that. And I think there's been, it really depending, again, what you mixed, you know, but there has been there is lots of jelly aids that are available on the market, and few studies have been made, and many do not impair the absorption of your drug. So I think it's on a case-by-case basis, I guess. What vehicle?
Yes, I guess so, yes.
But that's a very interesting question, because it creates a problem. I don't want to call it a problem, but ideally, for parents and carer, it would be nice to have on the SPC of the product the type of media you can mix your drug, your dosage form with. But then that creates, as you can imagine, on the other hand, on the industry hand, a very complex set of studies to run. So it's a double edged sword in a way, that it would be nice to be very be able to give a nice set of food or media you can mix your dosage form with for administration. But on the other hand, it kind of induce on the industry side a lot of study. So I think there is some work, at least within EUPFI, that colleagues are doing, trying to define a vehicle toolbox to be used during development, to be able to challenge your dosage form and then understand what will affect or not its bioavailability.
Yes, I agree that we miss some information in SPC, first of all. Yes, but this is complicated. Maybe another question. Yes, please, Professor Polak.
Thank you very much. That was excellent lecture. I have a question about the topic which you slightly mentioned during your presentation, which are cultural aspects. To what degree they influenced this field? And the second question is there something like a generally accepted good taste, regardless of the culture where the kids come from? Like, I don't know, McChicken or Coke or Snickers or whatever.
I would say that, and some studies are starting to appear. One, to answer your second point, one universal taste is milk until a certain age. And to answer your first question, there is a huge cultural divide, because again, as I try to identify it, we're used to what is available. And of course, we've got very different availability across countries, even within Europe. So there is a commonality, is that there is, so far, many pediatric products are liquid for obvious reason, because the dose is adaptable. However, that is as well a barrier to implementation of more novel dosage form. And in fact, I start to hear from industry colleagues that although mini tablets have shown a real potential in the studies that have been done towards facilitating their development and use with placebos, it seems that at market level, people don't like it. And I think it's just because it's so different. There is still lots of barriers, and they are cultural, they are perceived barriers. And I think maybe in 10 or 20 years time, the discussion will be very different. But right now we are at this transition where moving away from what the confidence zone, so giving liquids to children, is still difficult. And I've been living in UK for 20 years, where in fact you go to a pharmacy. I don't know where you're from, but here you go to a pharmacy. There is no suppositories. I mean, it's very few. And if you ask to your main street, your pharmacist for paracetamol suppositories for children, they will have to dig deep in their cupboards to find a box. And where in many other countries, to give ibuprofen or paracetamol for younger children, that is still the default position for preschoolers, for example. So definitely cultural or geographical, environmental are definitely very important.
Thank you, Catherine. We will close now this part of the session. I wish you good luck with your conference next week. Yes, and thank you once again.
Thank you for the invitation.
Hopefully we'll see very soon in person, and I would like to change a little bit the time of the next part of the session because we are a little bit late, so we will start 5 minutes to 5. So we will have almost 25 minutes of the break. Thank you for this part of the session. Thank you. We feel so pity that not all of you are with us, especially those who never visited Gdańsk. Hopefully we'll have another opportunity for you to invite to Gdańsk, which is really a beautiful city. Well, I will start the second part of our plenary session. I remind you that it's formulation concepts and delivery strategies for improving patients' compliance. And we as technologists, we will never make a good delivery system if we don't understand pharmacology and pharmacokinetics, and also physiology of gastrointestinal tract in the case of the oral delivery systems. So I have a great pleasure to introduce to you Maria Vertzoni, Professor of Biopharmaceutics and Pharmaceutical Technology from the University in Athens, National and Kapodistrian University of Athens. Professor Vertzoni is working on the impact of regional differences along the gastrointestinal tract and the influence on oral drug absorption. She is also in advisory editorial boards of the Pharmacy and Journal of Pharmaceutical Sciences, and what is very important for us, she is also a leader of one working group within the framework of ANGAP, cost action. This is a European network of understanding gastrointestinal absorption related processes, a very good network which contributes a lot in our understanding of drug delivery as oral formulations. I would like to ask you, Professor Vertzoni, for your lecture, Regional Differences along the Gastrointestinal Tract and the Impact on Oral Absorption.
Thank you for your kind introduction, and thank you for inviting me in this nice city. I hope to have the opportunity to sit tomorrow together with you. And my talk today will be, as Margaret said, in regional differences along the gastrointestinal tract and the impact on oral drug absorption. Doesn't work this. No, it's okay. Now it's on. Thank you. So part of my presentation, it's actually based on a review that we prepared 2 years ago with our colleagues of ANGAP, and it's an open access review in European Journal of Pharmaceutical Sciences, and it was the first trial of the group to publish on this, and I'm happy to share with you these findings. As Margaret said, ANGAP stands for Understanding Gastrointestinal Absorption Related Processes, and is a cost action that started a few years ago. And it's a network of 32 European countries with more than 500 members and plus 3 intercontinental partners. Our ANGAP has 4 working groups. The first one is focused on special populations, means pediatrics, geriatrics, and some parts of patients. The working group 2 focus on regional differences along the gastrointestinal tract. Working group 3 is dedicated to advanced enabling, bioenabling formulations. And working group 4 is dealing with food-drug interface. And of course, we have a dedicated work package for disseminating and also for exploitation and public engagements. So during these last years, we were able to organize 4 annual meetings, 6 training schools, and 2 workshops. And also, we delivered 19 webinars, and we prepared 3 videos that you can find them online, but also we prepared and published more than 15 publications in journals, and also we have a special issue dedicated for ANGAP in European J-Pharmacy. Please follow us on Twitter and LinkedIn. And our focus is the oral drug products, as the oral route is the most popular for drug administration due to patient compliance. And it's nice to see that more than 50% of new molecule entities approved the last years in the States are administered orally. So the focus is to find the appropriate in vitro and in silico tools in order to evaluate these products. But in order to be able to create good in vitro and in silicon tools, the knowledge of regional differences along the GI tract is of interest. So today we're going to discuss briefly how the GI mucosa, the anterior water layer, and also the intraluminal environment is changed because of the regional differences along the GI tract. And then we will present an example on the impact of regional difference on drug precipitation in the upper GI lumen. So if we go through the GI tract, we can start from stomach, go to the duodenum and the jejunum, and finally to the ileum and ascending colon. All these regions are of interest when dealing with oral dosage forms, and the last part of the colon is of interest also where developing modified release dosage forms or colon targeting dosage forms. So the knowledge of GI mucosa, anterior water layer, but also the intraluminal environment that the dosage form is going to face is very important. Starting with the GI mucosa, the GI mucosa is the innermost structure of digestive tract that influences absorption and secretion. The epithelial monolayer of the mucosa is in direct contact with the luminal contents and is important in regulating the passage of nutrients, ions, and solutes through paracellular but also transcellular pathways. In this figure, or your right-hand side, you can see here how the functional differentiation of the GI mucosa along the GI tract is, with different parts of cells, but also the surface area that changes in the small intestine. Oral drug absorption is influenced by intercellular tight junctions and the expression of transporters and enzymes in epithelial cells. On the other hand, the anterior water layer exists above the epithelium as a stagnant boundary layer of varying dimensions. The anterior water layer forms part of the barrier for molecules diffusing across the luminal phase into the cell. The surface layer on the top of the mucosa is slightly acidic to neutral. Then, as we go from the stomach to the small intestine, we can find that the secretion of gel-forming mucins occurs in a region-dependent manner along the GI tract, with up to now different identified 20 mucins. Gastric mucus is enriched with gel-forming protein, and on the other hand, mucus on the mucosa of the small intestine can limit the absorption rate and also the bioavailability due to chemical interactions between glycoproteins and the drug. When going to in vitro setups in order to study permeability of the API, the CACO2 monolayers are unable to support this acidic microclimate that has to be mimicked in the culture in order to obtain representative behavior, and this is a gap. Up till now, we described the mucosa and the anterior water layer, which plays a role for the API, for the active pharmaceutical ingredient for the drug. When we go and we would like to study the performance of the oral dosage forms, but also in a second phase also of the API, what is of interest is the intraluminal environment. This environment is dynamic, is complex, variable in terms of composition, given that we have constant addition but also removal of fluids in the GI compartments, and the physicochemical characteristics of the GI tract change not only with the region but also with the dosing conditions. And based on bioavailability and bioequivalence study, we can identify two states of administration, two conditions of administration. The first one is the fasted conditions, where healthy volunteers arrive into the clinic after an overnight fast of at least 10 hours, and then the oral dosage form of interest is administered with a glass of water. On the other hand, in the fed state conditions, as defined in food effect studies by FDA and DMA, it's a little bit different. These conditions are described as following an overnight fast of at least 10 hours, as it was the case for the fasted state conditions, and then the study subjects should start the recommended meal 30 minutes before the administration of the drug product. And these meals, as you can see in the figure, consist of 2 fried eggs, 2 toast fried also, 2 slices of bacon, potatoes, and milk. This means in total a high fat, high calorie meal, in terms of calories is about 1,000. And then 30 minutes after the initiation of the consumption of the meal, the volunteer can take the dosage form together with a glass of water. It should be noted that these conditions induced by fat, high-calorie meal provide the greatest effect on GI physiology and the maximum effects on the systemic availability of the drug, so it's easy to identify the food effects that can be because of the administration of the product. Nowadays, in the new guidance, the FDA try also to insert the moderate meal that has approximately 500 calories, but it's in infancy yet. And based on these two extreme conditions, we try to identify the intraluminal environment and characterize it in terms of pH, buffer capacity, osmolality, total bile salts contents, short-chain fatty acids, cholesterol, phosphatidylcholine, total protein content, and total carbohydrates in order to understand the environment that phase an oral dosage form upon ingestion. In order to do this, we invite healthy volunteers, and then we incubate them via the nose or via the mouth with a double lumen tube, and we're able to administer the water or the meal via this tube directly in the stomach of healthy adults, and then we can aspirate samples from the stomach or the upper small intestine, in the case that we're interested in the upper lumen, and then via colonoscopy we can aspirate samples from the ileum or the ascending colon. And in this figure, we can see how these fluids look like. In the upper panel, we can see contents of the upper small intestine aspirated 5, 10, 20, 30, and 40 minutes after the administration of a glass of water. And the lower panel, we can see initially how the contents of the ascending colon looks like when we collect them, and how they looks like after ultracentrifugation, when in the upper part we have the aqueous phase, and the lower part, the solid part, that is the bacteria that are present in this environment. We're not going to point out this topic now, because it's a huge topic, how bacteria can affect absorption, because they can degrade the API or the prodrug, so it can have a positive or a negative effect on absorption, but we will not touch this because it's another story and big story, but very interesting. So if we go to the table to see how the pH changes across the intestinal lumen in the fasted state, we can see that we start with a pH of 6 to 7 in the upper small intestine, and this pH goes to 8 at distal ileum, and it remains high at cecum and ascending colon at values at around 7.4, 7.8 as median values. This is very crucial to know for modified release formulations which have polymers in order to protect them, and how this polymer can be released in the lower part of the intestine in order to deliver the API. In terms of buffer capacity, the buffer capacity increases as we're going down to the lumen, and the pH together with the buffer capacity is a characteristic that can play a major role when we have to deal with the compounds that are lipophilic and ionizable. Also, the osmolarity decreases as we go to the ascending colon. The contents of distal ileum, cecum, and ascending colon are hypoosmotic. And then we have that content in terms of total bile salts. Bile salts are present even in the fasted state in the small intestine, which together with phosphatidylcholine can create micelles and enhance the dissolution and the solubility also of lipophilic compounds. Also, cholesterol and phosphatidylcholine are also present in the ascending colon, although when we started the studies, we didn't expect to receive so high values there, even higher than in the upper small intestine. And we have not to forget also the presence of short-chain fatty acids in the ascending colon that are produced because of the presence of the bacteria. On the other hand, when we go to the intraluminal environment in the fed state, that means after the administration of high-fat, high-calorie meal, we can see how these samples from the upper small intestine of healthy adults looks like in the upper panel, from half an hour up to 4 hours after the administration of the meal. And we can see how the viscosity changes, how the presence of solid states half an hour after the administration of the meal, and how this returns almost to the fasted state at the last sample, where the sample is more clear. And also in the colonic samples, that is the lower part, we can see that the color seems the same as before in the fasted state, but here the presence of solids is bigger, and also the presence of bacteria can be changed in the fed state. And here in this table, we have the physicochemical characteristics of liquid contents along the intestinal lumen. Here, it's nice to notice that although in the upper small intestine, the pH value is around 5 to 5.5, in distal ileum remains 8. So more or less in distal ileum, in the fasted and the fed state, the pH is the same. On the other hand, in cecum, the pH is lower, is 6.4. It remains at about 6 at the ascending colon. Buffer capacity is higher than the fasted state because of the presence of nutrients, and the ions because of the ingestion of food, and buffer capacity again increases as we go to the ascending colon. And another component that has huge differences compared to the fed state is the total bile salt presence because of bile that secretes bile salts in order to digest and the meal that there is there together with lipase. So based on these findings, we would be able to create in vitro tools, but always you have to keep in mind the impact of gastrointestinal variability on oral drug absorption, which this variability gives us also the variability, contributes to the variability also in the pharmacokinetic profiles. And this year, through UNCAP again, we were able to publish another review on impact of GI tract variability on oral drug absorption and pharmacokinetics. But this is not the topic of today, just for your knowledge, is an open access again review. And now we'll go to an example of the impact of regional differences on drug precipitation in the upper GI lumen, based on the findings of in vivo data. First of all, the intestinal supersaturation and precipitation can occur when the drug is in solution in concentrations higher than the equilibrium solubility. Please keep in mind that it's different, the supersaturation versus the solubilization. Supersaturation is a state that is thermodynamically unstable, is a situation that lasts not for more than a few minutes maybe, depending on the API. Solubilization is a stable situation. So when the API or the dosage form that we administer can create supersaturated concentrations, this means that the plasma levels are higher than anticipated from equilibrium solubility data. As the supersaturation is an unstable situation, this drives to precipitation, and the precipitation can reach a concentration close to the equilibrium solubility, but sometimes if the state, the crystalline, for example, versus the amorphous form of the API is different in terms of solubility, then we can have concentrations even lower than the solubility of the API in the dosage form. And this can lead to decrease in early exposure. This means decrease in peak exposure, but also increase in variability exposure because it depends on how long the supersaturation will last. And this, on overall basis, will impact the efficacy, but also the safety of the drug. And if you want to study this in vitro and see the impact of regional differences on drug precipitation in the upper GI lumen, this has a very high interest in the fasted state because drug precipitation can occur in this region, mainly when we have the administration of poorly water-soluble bases, given that in the fasted state, the pH in the stomach is expected to be 1.2 to 2, but, and here a weak basis can be in solution, depending on the Ka, but then upon entrance in the upper GI, in the upper intestinal lumen, here with the duodenum, the pH is 6.2 to 6.8 approximately, so precipitation of the weak base, lipophilic weak base, is expected. The same can occur also for enabling formulations where we have amorphous solid dispersion, for example, which can precipitate in the small intestine. In order to study this, we create an in vitro system in our lab. We call this a BioGit system. The BioGit stands from Biorelevant Gastrointestinal Transfer System. And this was created with the aim to evaluate drug concentration in the upper GI lumen after oral administration to fasted adults. So it's of interest, especially when concentrations could be affected by potential precipitation in the upper GI lumen. This setup, it's from commercially available equipment. We use commercially available mini vessels from Erveka and mini paddles from Erveka and Distec. So we have a three-compartment model. We have the gastric compartment, the duodenal compartment, a reservoir compartment, and based on luminal data, we have calculated this flow rate from the gastric compartment to the duodenal compartment, and all these flow rates are based to a half-life gastric emptying of 15 minutes. The paddle is rotating to 75 RPMs, and all the equipment is maintained to 37 degrees. In the reservoir compartment is used in order to maintain constant the concentration in the duodenal compartment. In order to have biorelevant conditions, we use biorelevant media. So in the gastric compartment, we use the FASSGF. FASSGF stands for fasted state simulating gastric fluids, and we start with initial volume of 250 ml. And in the duodenal compartment, we have the fasted state simulating intestinal fluids and the constant volume of 40 ml. So, in the reservoir compartment, a series of phosphate buffer solutions containing sodium chloride, bile salt, and lecithin are pumped from the reservoir compartment into the duodenal compartment in order to maintain the composition of simulated duodenal contents, that means the pH, the buffer capacity, the osmolarity, the bile salt, and lecithin concentration unaltered during the experiment. Please note that in vitro conditions that I described in the previous slides are based on volume of duodenal contents and input and output duodenal rates estimated after modeling luminal data of high permeable drugs. Luminal data collected from 12 healthy adults after the administration of weak bases, ketoconazole and dipyridamole. The conditions in the duodenal compartment take into account both the transport of a high permeable API via the epithelium of the upper small intestine and also the transit through the upper small intestine. And this in vitro methodology complies with the continuous GI transfer process that occurs in vivo. Using this system, we're able to estimate the percentage solid fraction of API that is available in the upper small intestine, but also we are able to estimate the degree of supersaturation. In order to do so, we can sample from the exit port of a BioJet and then estimate the total presence of the API in this region, and then through filtration, we can measure the dissolved API, this means the concentration that's available for absorption, and then an excess amount of the drug can be pre-weighed, and we can add there the solution that exits BioJet, so we can estimate the equilibrium solubility in each time point. From the concentration and the equilibrium solubility, we can measure the degree of supersaturation in each time point. As an example, I will show the results for fenofibrate when it was tested in the form of solid dispersion capsules with different doses, 54 milligram and 160 milligram. Fenofibrate is a neutral API with a log P of 5.3, and we use these different dosage dosing conditions for solid dispersion capsules. And in this figure, we can see how the apparent fenofibrate concentration in the duodenal compartment of BioJet versus time are for the two different doses. The low dose with blue symbols and the high dose with yellow symbols. With gray symbols, we can see here, for both APIs, the mean equilibrium solubility values in the contents of the duodenal compartment. So we can notice that contents of the duodenal compartment are supersaturated with fenofibrate. If we want to have a flavor of this impact on oral drug absorption, at least for the early exposure, that means what happened 1 hour after the administration of this API, we used the plasma data from the clinical study, and we calculate the partial AUC from 1 hour after the administration of these tablets to healthy adults. And here are the partial AUC values, the plasma data, And in this row, in the gray row, we can see the biology data. That means the AUC that we can calculate from our data. What we can notice is that differences of AUC 1 hour after the administration of this product, phenylfibrate product, between the two doses were statistically significant for the two doses. And this applies for the plasma data, but also for BioJITS data. And what we can see also is that the ratio of the early exposure of this AUC are similar both in vivo and in vitro. So the in vitro setup can give us a prediction of the performance, the difference performance between the two dosage forms. But also, BioJIT can inform PBK modeling on precipitation kinetics in the upper small intestine. Usually, PBK modeling uses precipitation characteristics, precipitation time, in order to model for the modeling purpose of PBK. So precipitation time in this example, the example of a compound that it was a semi-fumarate crystal and administered in the form of capsule and at the dose of 100 milligram. This compound was a weak base with a log P of about 3. The precipitation time was estimated by modeling BioJIT data into gastroplasm. Firstly, an effective mean particle size was fit to the solution data generated in FIRES SGF in order to estimate the input dissolution kinetics for the system. So we calculated the particle size, and then using the advanced compartmental entrasion model, the CAT file in gastroplasm, we generated different profiles in order to simulate the BioJIT system. And as we can see in this figure, using gastroplasm, we could simulate different lines that represent the concentration in the duodenum of healthy adults. And we compared this with the observed data in BioJIT and tried to fit and understand which precipitation time is more appropriate to use in the modeling. By default, gastroplasm uses a precipitation time of 900 seconds, but as we can see, longer precipitation times of about 3,000 or 10,000 seconds predict both the initial phase, the 15 minutes, but also the 45 minutes time points. And if we go to plasma data, initially with squares we can see the observed data, but let's see how the plasma profiles looks like when different precipitation times are applied. And we can see that precipitation time can play a role when predicting, when simulating the plasma data in healthy adults. So we need this information. And if we're going to highlight the observed data, we see that indeed, longer precipitation times allow for a Cmax prediction within variability of the clinical data. The impact of regional differences in anatomy and physiology along the GI tract on the absorption of drugs is profound, and understanding the nature of the barriers is challenging. And the knowledge of regional changes in the properties of the GI tract are an essential component of mechanistic models to predict drug absorption and are particularly important to guide development of oral dosage forms. These opinions and all the gaps that we have to fill now are summarized in another paper that it was recently published in Advanced Drug Delivery Reviews, again from ANGAP network. And today we discussed briefly how this environment is in healthy adults, what happened in special populations, what happened in geriatric patients and older people. We're happy that last January we started a training network in order to understand what happened in geriatric population. And this is HPOP. HPOP stands for absorption in geriatric patients and older people. And the HPOP aims at generating for the first time information on the actual dosage form intake conditions for older people and geriatric patients and on physiological characteristics of their GI tract relevant to oral drug absorption. It's a program that was funded by Horizon 2020 under Marie Curie Action with a project budget of 2.8 million euros. This will last the next 4 years, and we're happy because we're a consortium of 14 organizations for 5 European countries. We're 3 universities that are participating, KU Leuven, University of Gdańsk, and National Kapodistrian University of Athens, which we are the coordinator. And we're happy to have with us a lot of FPA partners all over Europe in order to try to investigate this new topic, this new information. But our aim is not only to doing research, but also to train new ESRs, new early stage researchers. And in this program, we have 11 new PhD students who are going to work, I hope with passion, in this program. And I hope to another meeting to present you some findings from this consortium. I would like to thank you for your attention, and I would be happy to answer your questions.
Thank you, Professor Vertzoni, that you helped us to understand more about gastrointestinal tract. And I always claim that we know more probably about Mars and other planets than about this gastrointestinal tract. So the lecture is open for discussion. Is there any question? Yes, Professor Breitkreuz.
Thank you very much for the interesting talk. We had a project with a similar API as phenylfibrat, which was even less soluble, and we had significant difficulties to distinguish between the precipitated drug molecule and the drug molecule in solution. So my question, in such a transfer model as you have had here, so how did you do that? To precisely say this is precipitated part and portion, this is a soluble one.
Thank you for your interesting question and challenging question. It's something that always is difficult to know when we have amorphous solid dispersion and nanoparticles. That's why it's not easy always to deal with nanoparticles and with great certainty. Usually what we are doing is that we use filters with very small pore sizes. We pre-filter with 0.45 or 0.2 filters, and then we go for nanotop filters which have a particle size of 0.1. In some cases also we are doing ultra-centrifugation in order to be sure that we can estimate with accuracy the percentage of this part that is in solution. That's why, as you may have seen in the figure, we mention apparent concentration and not concentration.
Another question? Professor Polak?
Sorry, thank you. First of all, great lecture. I'm really looking forward to the data which we'll generate during this project. I'm crossing my fingers for you. Very detailed question. On one of your slides, you presented the AUC ratio, AUC for the plasma concentration versus AUC from the BioGit tool. In the plasma, it was 0 to 60 minutes. In the BioGit, it was 0 to 45 minutes. Do you think it can influence the result, and why there was this?
The problem is sometimes that in the clinical studies, we don't have enough points in the early phase. So we have just a few points, and the estimation of AUC is not always more representative. So sometimes we prefer to have 0 to 60 in order to not to have just 1 point, to have a triangle, but to have an AUC. In terms of BioGit, 45 is the sampling, but this sampling is the midpoint, so maybe more accurate is to put 15 minutes. So it's from 0 to 15 minutes, the AUC calculated, and we think that this 10 minutes difference is not expected to play such a huge role in the differences in the AUC. Yes, because it tries to precipitate. No, we just cut, yes, because we don't know exactly how it goes. It's not so, if I can go back to the slides, please. May I go back to the slides? Is it easy? Thank you. Because it's not easy to extrapolate when you have BioGit experiments. It's not like plasma that you expect to go to 0. Usually when you have biojet, you expect to go to solubility, to equilibrium solubility values, but in order to be more reproducible for both APIs, we selected to cut exactly at the type point that it cuts. If you want, I can show you.
There is another question, please introduce.
Yes, first of all, thank you very much for the excellent presentation. I have seen the data of the BioGI model. It's really very interesting and show clear correlation between the in vitro and in vivo data. Excellent. At least regarding the dose, if you compare the dose, we see this correlation clearly. The question is this model discriminating enough to see differences in different formulations?
Yes, thank you for the question. I just selected one example because I wanted to point out on the absorption, as this was the topic of this conference. Initially, we have performed experiments comparing the data that we collect from BioJet, the duodenal concentrations, versus intraluminal concentrations, that have been measured in healthy adults. And in this case, we have examples where, for example, Sporanox solution, that it's a cyclodextrin solution, was administered in comparison to a solid dispersion. So we were able to identify, to measure and be within the in vivo data, intraluminal data. That's why, firstly, we have evaluated BioJet versus intraluminal data collected from humans, and then we have performed different experiments from and comparing the early exposure, as the example that I showed you, with different type of formulations, comparing type of formulation, and also comparing the different doses. And we were able to create a graph, and they have in the one axis the ratios from in vivo data, and the other axis the ratios for in vitro data, and this seems to correlate well. We have this in the publication that I showed, but I had not enough time to show you, but if you want, we can discuss later. Thank you for the question.
Lovely. I would love to see this. Can I have another question? So we since a long time we are discussing about the digestion process. If the digestion indeed take place only in the duodenum, yes or no? And up to now, at least, I don't have a clear answer for this. If I look to the data what you have presented, the samples from the gastrointestinal tract, it looks like, at least if I look to the bile salt concentration in the small intestine, and it's very high in the fed state, between 8 and 20 millimolar. And if we go to the ileum, it's very low. So the question is, how is the analytical method used? Is the analytical method able to distinguish between intact or the bile salts, only the free bile salts or all bile salts?
Yes, thank you for the question. In terms of digestion, it is stated in the references that the digestion starts in the stomach. It's not clear how much digestion occurs in the stomach based on armandetol and herneltol, three papers from two groups that have worked a lot with this, with in vivo samples. They argue that digestion in the stomach, is also the variability, can start for 5 to 40% of digestion can occur because of the gastric lipase. Now, in terms of bile salts, at least the results that I showed you, it was the measurement of bile salts in the aqueous phase of these intraluminal fluids, and this measurement was done not with a commercially available kit, but with HPLC, because in our lab we developed a method where we were able to measure also the individual bile salts. And with this method, it's not an MS, it's a corona, it's a detector that it's close to MS, but not so expensive as MS. But we were able to measure 10 different bile salts in the small intestine and also the colon, because in the colonic area, it was primary cholic acid and not the conjugated ones.
Thank you.
Thank you, Professor Vertzoni.
Thank you.
I would like to ask for the second lecture, Professor Sebastian Polak. Professor Polak is the head of Laboratory of Pharmacoepidemiology and Pharmacokinetics in the Jagiellonian University in Kraków in Poland, and he is a leading scientist in mathematical modeling of the biological processes and especially those concerning pharmaceutical approaches. So thank you, Professor Polak, for accepting our invitation, and the podium is yours.
Thank you so much. First of all, it's a great pleasure to be here. Thank you very much for the invitation. It's a great honor, really. Gdańsk is fantastic. Medical University in Gdańsk is probably, no doubt, one of the best universities in Poland, so it's really a great pleasure to be here. I'm actually wearing two hats. I'm a university professor, and I'm a scientist working for Certara UK, and the materials which I'm going to present today is a mixture of work which we do both places, actually. I will start with the apologies, because when I was going through the slides this morning, I realized two things. So the first one is I am going to talk about the modeling, mathematical modeling, and there is no single equation. So how to discuss mathematical modeling without equations. And the second thing is I prepared too much material and too many slides, so apologies. I will storm through some of them at least, just to make it quicker. So let me start with the apologies. If only the remote controller worked, I would do that. Doesn't work. Oh yeah, so apologies. Let's start with the apologies. So let's start with those equations. Apart from this slide, there will be no equations whatsoever at all, really. But I want to prove that I'm really going to talk about mathematics. So all these equations are there, but hidden at the background. And just to prove it further, there's a screenshot from the recent manuscript, which we have just submitted, really. And as you can see, there is equation number 80 and 81. There's almost 100 equations in this paper. By the way, it was one of the most painful scientific adventure in my life. Writing this paper took us 6 years almost. It was really a pain. But anyway, there is mathematics there. I promise, even if I'm not showing the equations. A little bit of an introduction, theoretical introduction, sorry about it. But I promise I will go into the case studies, real life case studies. So I'm going to talk about the PBPK, which is physiologically based pharmacokinetics, and the utilization of PBPK in the real life, or how to tackle real life problems and try to solve them with use of PBPK models. However, I would like to show where this PBPK fits really. It's part of a larger paradigm or idea or approach, which is called MIDD, which is model-informed drug development. It's a combination of knowledge, expertise, tools, data, and mathematical modeling. And PBPK is one of the elements of this MIDD, which is very practical. It really helps to solve certain problems, and it is something what is not a science fiction anymore. When I started 15 years ago to work on that, it was partially at least science fiction. Right now, it is purely science. There is no fiction there. So what is PBPK in general? It's a mathematical framework which allows us to describe certain processes with use of the mechanistic models, mathematical mechanistic models. And based on that, and because of that, it allows us to dissect each and every problem into three parts. The first one is systems data. The second part is drug data, and the third part is trial design. So everything what influences the fate of the drug, but it's not the drug itself. Dose, dosing, dose staggering, and so forth and so on. So with use of this paradigm and this dissection, we can really try to tackle different type of problems, issues, and try to solve them, as I will try to show. So what kind of the input data we need from the drug part, from the drug perspective? We have to describe API with use of Physchem data, with use of the ADME data, sometimes with use of the PK data. The kind of a holy grail is to do a real life IBE, which means to extrapolate from the in vitro situation to the in vivo situation. So to take the in vitro data and to try to extrapolate it to the in vivo situation. to predict what will happen in humans without even touching humans as part of the clinical trials. That's the holy grail. It happens, it is very challenging, but it's doable. In most cases, what we do is called middle-out approach. So we take everything what is possible, and we try to use this knowledge to extrapolate it further and to answer or to explore unknown areas. So we do take, apart from the Physiochem and ADME data, information about the PK, which has been generated for the drug of interest, sometimes early stage type of clinical trials, or some other sources of information which can be used. And then from the systems data, we need the information about the human body, in fact, because the system of interest is human body here. And there is a question which I hear very often, so how much data do we really need, or how many parameters we have to consider? There is no straight answer for that. It depends on the question, it depends on the model. Sometimes there are 3 parameters, sometimes 300 parameters. It really, really depends. But in general, we want to describe the system, the human body, as detailed as possible to feed this mechanistic model which we have depicted here. And at the end of the day, the endpoint of interest, what we're really interested in, is the fate of the drug in the human body. So what is the concentration in different parts of the body, not only in plasma, blood, but in the different tissues as well. What is the acceptance of this kind of approach? And that's something what I love, and it's really absolutely fantastic. It is good and it's growing. And as good, I mean regulatory bodies are accepting PBPK models, and they're accepting utilization of these models to waive clinical trials. So instead of running 20 clinical trials, we can run 10, and the remaining 10 can be simulated, which is absolutely fantastic. Of course, it's not the case for each and every situation. We have to be aware of that, because we don't know the system very well, because we don't know the phenomena which occur in the system very well, because we don't know what will happen in different populations. It was mentioned by Maria earlier on. So there are those specific or special populations which are very challenging, but at the same time to model, but at the same time they're even more challenging to explore in a form of the clinical trials. Let's imagine the clinical trial in the pediatric, in the neonates, clinical trial in pregnant women, in elderly people, and so far and so on. It is really challenging. With a properly developed PBPK models, we can tackle this kind of problems, and we can try to solve certain issues which are connected with this kind of situations. So what I'm going to focus today on is dermal application and dermal absorption of the drug. So the model which I will present is a model which follows the paradigm which I have just described. So it takes the information about the system, it takes the information about the drug, and in the, say, scaffold on the trial design allows you to assess what will happen with the drug when it is applied topically. What will be the penetration of the drug? What will be the concentration in the local tissue? What will be the concentration in the body, in the plasma, so the systemic concentration. This kind of, let's say, combination of data or structure of the model allows us, by modifying those system-dependent data, simulate the situation in those specific or special populations which I mentioned previously. So I will go very quickly through some of the special populations. I will show you some of the data which were collected for certain parameters, not all of them, it's just an example. But hopefully it will give you a taste or flavor of what can be done really. So let's start with the disease population. In this case, it's a psoriatic skin or psoriasis. And what will be the important skin parameters which can potentially influence the behavior of the drug? What changes as part of this disease or as a cost of this disease? The pH of the skin changes, decreases, sometimes really significantly. Temperature of the skin changes. The stratum corneum, which is the main barrier protecting us from the xenobiotics to be absorbed through the skin, is modified. So the thickness changes, number of layers changes, the structure of the corneocyte changes. In general, the stratum corneum is thicker, but at the same time, there are cracks. So what will be the influence of those crux on, for example, a drug which is applied topically on the disease-modified part of the skin. All of that has to be considered, and each and every of those, how we can see that, red colored elements or parameters has been described as part of the model which I'm presenting right now. We go deeper through the skin, to the viable epidermis, dermis, up to the subcutis, and there's more and more parameters which we have to consider and which we have to modify if you really want to simulate disease population as opposite to the healthy human skin. Even more challenging situation is with the pediatric population. Why? Because we do have to consider one thing which is crucial, ontogeny. As it was mentioned during one of the previous presentations or lectures, 3 days old newborn is very different as compared against 4 or 6 or 7 days old newborn, because certain physiological parameters, anatomical parameters changes really quickly. And that's something what we have to consider. The same applies to the skin. So for all the drugs which are applied topically, we have to consider that. So pH of the skin changes very quickly. Stratum corneum thickness, viable epidermis thickness, all of that really changes. And to be able to describe this population mathematically, we do have to provide this data, quantitative data. So we do have to measure it first to understand the system to be able to simulate it later on. But obviously, it's not only about the skin, and that's something what we have to keep in mind as well. So even if we are interested in the topically applied drug formulation, it's not necessarily only the skin which influences the pharmacokinetics and pharmacodynamics of these drugs. Simcyp level changes significantly. Amount of the metabolizing proteins in the liver changes significantly with age. The same applies for plethora of other parameters. For each and every of this parameter which we know about, we have to collect the data or generate the data. That's why I'm so looking forward to the results of the study which Maria mentioned previously, because there are gaps. We know that. And we are really looking forward to this kind of information to be provided. It's not that super easy as you can imagine. The same with the geriatric population. Certain parameters changes with age. By the way, small digression here. When we started this project, it was 6 years ago, 2014, 2015, when we received a grant from FDA. We went through the literature trying to gather data which describes how the skin changes with age. And it was kind of obvious that skin changes because everybody is talking about that, and it's kind of an obvious. There is surprisingly small amount of information, quantitative information, how the skin really changes with age. It's just like common knowledge. I can see my skin is different than 20 years ago, but how different in terms of numbers? It's not that super easy to find this kind of data. At the end of the day, there's not so many parameters which really changes. There are structural changes which are kind of visible, like, I don't know, the composition of lipids, which are part of the stratum corneum changes, or keratin level. But it's not that super easy to describe with the use of the mathematical language. Anyway, there are some parameters which change. But at the same time, again, the same situation which we had with the pediatric population. It's not only about the skin. There's a number of physiological parameters which change with age. Simcyp level, kidney weight, and amount of the metabolizing protein, so the excretion will change and so far and so on. We do have to consider all of them to be able to precisely predict what will happen in the geriatric population. I'm going to go through the details. The same applies for other populations, so-called specific or special populations like liver insufficiency or kidney insufficiency. For each and every of this parameter, we have to find the data. We have to define what is the change connected with the severity of the disease, sometimes in combination with age, and to be able to provide that. So in general, and it's one of the criticisms which we hear very often, PBPK models are data hungry. That's true, but the good thing is, once you have it, you can use it for different problems, for different virtual scenarios. So yes, developing a, let's say, database or this, nicely or not so nicely looking graphs. I hate 3D graphs, sorry. Takes a year to combine them together to analyze them for a group of 2, 3 really capable scientists. It looks simple, but it's not that super simple. But once you have it, you're pretty much done. You can take this data and you can reutilize them multiple times. Okay, as I said previously, I'm going to focus on the dermal absorption and to give you real life examples for the special populations and the absorption of the topical application. So what kind of data we need? We need information about the API, log P, pKa, protein binding, partition coefficients, and the diffusion through different parts of the skin. That's something what needs to be either measured for the API of interest, or because some of them are really not that super easy to be measured, they can be predicted with use of the mathematical models, in this case QSR models. So we have to provide this data for the drug of interest to feed the model and to be able to run the simulation. So that's our input, which we provide to the model. But apart from that, and that's something what we can't forget about, and that's why we all are here at this conference, at this meeting, we can't forget about the formulation. And that's something what is not that super trivial for modelers. It's not that obvious for modelers. Drug formulation does matter, and information about the formulation is absolutely crucial to understand the fate of the drug after the application, regardless of the route of application of the drug. So generally speaking, when we think about a dermally applied drug, there are 4 different types of groups of families of formulations, beginning from relatively simple solutions through more complex emulsions. The situation becomes even more complex when in the emulsions there are solid particles, suspensions, up to the patches or transdermal therapeutic systems. If my student said patch or plaster in Polish at the exam, he, she would fail. So no patches, TTS. So once you know your formulation, you have to characterize your formulation. So you have to understand how this formulation behaves. So we need information about the evaporation, information about the diffusion of the API inside the formulation. You need to characterize the rheology of the formulation to be able to provide this data to the model and then to predict the fate of the drug. The model which I'm going to use today is a so-called MPML McDermott model. It's a model which we've been developing for the last 10 years almost 10 years, yes, beginning from 2012, that was the beginning. It's a PBPK model which covers or follows the paradigm which I have just described. So drug-specific data, formulation-specific data, information about the population or the system, in combination with full PBPK model which describes the human body, it allows us to understand what will happen with the drug when it's applied topically. Some part of the information which can be used as an input comes from the in vitro studies, or ex vivo studies to be precise, in this case from the IVPT study or in vitro permeation study. So we can use this kind of results to feed the model and to predict what will happen in the real life situation in vivo in human. However, after certain time of using this data, we realized that it is possible to not only use this data here, but we should try to simulate this data. So let's try to simulate the in vitro system to be able to understand in a relatively simple system, which is a part of the skin in the front cell, regardless of the technique, if it is static flow or static or flow through system, doesn't matter. Let's try to simulate this system and extrapolate it farther to the in vivo situation. So instead of running the in vitro study, let's try to simulate this study, because it will allow us to modify certain parameters to be able to understand what will happen later on. So actually what we are following here is a kind of a holy grail of PBPK modeling. We do IVE or in vitro in vivo extrapolation. So we take the relatively simple data, physical-chemical parameters, Q1, Q2, Q3, description of the formulation of interest. Here are some examples of those Q3, like particle size, rheology, viscosity, these kind of things. We simulate the in vitro experiment. Then we take the parameters like diffusion coefficients, partition coefficients. We take it farther to the in vivo situation, and we see whether it covers really what we can expect in the clinics. We're comparing versus the observed clinical data. If the prediction is good, we are done. We've got a good quality model. We can take it farther and use it for different populations, those special populations. If the results are not that great, we are coming back to the in vitro model, relatively simple, both mathematically and conceptually. We modify certain parameters. We check which of the parameters, influences the behavior the most. Then we are coming back to the in vivo and so forth and so on. It's like a circle of learn and confirm. Once we are done with the model development, we can take it farther and we can really utilize this model in the real life. And I've got two case studies. One of them is really practical and really taken from real life. The other one is a bit more theoretical. Depending on the time, I'll definitely go through the first one. Let's see what I will have enough time to go to the second one. So the first one was a collaboration between Galderma. I can say that because it's a common knowledge, and Simcyp or Certara UK. The sponsor in this case, Galderma, was interested in using PBK model for certain very specific problems or to solve the problems. So there were two types of problems and two aims of building and utilizing this model. So the first one was, by the way, I've stolen those slides from Dr. Wagner presentation from APS in 2019. She agreed to that. So there were two aims. The first one was to run drug-drug interaction study for trifarotene, a cleave cream, versus fluconazole, which is a moderate CYP inhibitor, 3, 4, and 2C9. And the second, even more challenging, was to lower down the acceptance level for the age, so when the drug can be used. Because in the label, it says from 12 to 17, it is acceptable because there were clinical trials run for this age group. But the company wanted to lower it down to 9, 11. And the challenge was, or they went to FDA, and FDA officers said, yeah, that's fine. You have to run the clinical trial. Galderma said, okay, we can do that. They started recruitment process. After 2 years of recruitment, they found one volunteer whose parents agreed to take part in this trial. There is no much of the statistical significance after study run with one individual. So they came back to FDA and said, we have a problem, we can't do that. So what we should do? And the FDA said, consider PBK model. So they came to us, and my colleague, Dr. Patel, developed the PBK model with use of the scaffold, which I have just described, this MPM-MEC-DERMA model. And he used the data which were available from the sponsor. So information about the physicochemical parameters of the trifarotene, very detailed CYP profiling, information about the transporters, plus very useful stuff, which is the RAT radioisotope tissue distribution study. So that's really something that helps to have a good quality PBK model. And then we ran these two arms of the study. So the first one was drug-drug interaction study with adults, or with adult population, if you like, healthy individuals. That's good enough for regulatory bodies at the moment. So we virtually, with use of the model, applied both drugs at the same time. Topically applied trifarotene and fluconazole. And the therapeutic dose of fluconazole gave about 20% of the inhibition of the enzymatic activity of CYP2C9 and 3,4. And then we asked the system, what will happen clinically? What will be the effect? How the exposure will change after this combination given concomitantly at the same time. So what you can see here, this green line is the result after several doses of both drugs without, in the first case, it's without fluconazole, and this red dotted line is with fluconazole. So what we can see here, what is the conclusion out of it? There is an increase in the exposure. However, the increase is far away from any clinical significance. We're at the level of the quantification. It's far away from the toxic dose and toxic exposure, so it's safe. But then we ask the question, what will happen if the drug will be inappropriately used? So in case of, I don't know, small area of the skin, there is a huge area. So this teenager, 10 years old guy, took a cream and put the whole tube on himself or herself, plus 3 tablets of fluconazole. So purely, let's say, theoretical virtual scenario. What will happen then? So what we did, we actually blocked 100% of the inhibition capacity of CYP2C9. And we asked the system what will happen then. And the result was pretty similar. There will be increase as compared against the baseline. The increase will be significant, but it's still far away from the clinical significance. We obviously described it very nicely. It was a huge report, 300 pages long report, and the sponsor took it to FDA, and the FDA response was, we accept that. So instead of running clinical trials, we built the model, we ran the model, and FDA accepted that. It's not that uncommon, actually. There are about 300 drugs right now on the market which have PBPK models as part of the label, or results of the PBPK models as part of the label. But to my knowledge, it was the first one for the topically applied drug, which was a big thing actually for my group in Simcyp. And the label says no clinically significant differences in the PK of trifarotene were predicted with PBPK model when used concomitantly with fluconazole. So kind of a success. So that was the first arm of the study. But then there was the second arm. Can we really lower down the age of the kids which can use this drug without running clinical trials? And that was a real challenge, because first of all, we had to develop the model for the pediatric population. We had to verify the model with the use of multiple drugs, not only trifarotene, because FDA wanted to see, the whole set of data and verification of the model, and then verification at the level of the trifarotene, and then the real life like simulation. What I'm showing right now is just the final result of the simulation. It took months to do all of that, obviously. It's nice to show that in form of the one slide, but it took really months to generate this slide, or the content of this slide. So what we can see here is a comparison of the observed data for the group of 12 to 17 years old adolescents. The prediction of the model for this group, for this age group, which was acceptable. So we covered the clinically expected plasma concentration nicely. And then we lowered down the age, including all the modification of the skin and the system, which we can expect from 9-11 as compared against 12, 17 years old kids. And there was an increase because the barrier is different, because the metabolism of the drug is different, because of all the other phenomena which occur in the body. However, this increase was not significant. We're still at the level of the quantification. And again, it was taken to the FDA, and FDA accepted that. So it's part of the label right now. So instead of running clinical trial, we built the model, we developed the model was accepted, and it's part of the label. And I trusted to the level which, let's say, allowed me to take and use this drug for my daughter. I used this drug for my daughter because I trust in what we did. It didn't work, by the way, but it's a different story. The second case study. I still have time, so I'll try to go through that very, very quickly. It's a bit more theoretical, so there is no, let's say, communication with regulatory bodies here. It was more for our understanding of the system and the question which we can ask the system. So it was selegiline and its metabolites and different formulation. In this case, it was patch or TDS. And the aims of this study was to test whether we can predict the fate of the drug, including metabolites, 3 active metabolites, a pretty complex situation, to assess transdermal drug penetration and the metabolites exposure after this application of the patch for different individuals, for different populations. The same model. So the scaffold is the same, and that's the beauty. Scaffold is the same, MPML McDermott model. The input was relatively similar as for the, let's say, shape, but obviously the values were different because there is a different drug. So PhysChem data, ADME data, rather precise and detailed metabolic characterization or metabolic activity characterization for this compounds of interest, and information about the release from the patch, because that allows us to, predict what we can expect in terms of the concentration of this drug in the plasma. So what was interesting here, and what's the difference between the previous case study and this one? In the previous case study, we're interested in the local tissue concentration mainly. The plasma concentration is kind of an off target. We don't want the drug to go to the plasma. We want the drug to act locally in the dermis, in acne. However, in this case, it's separate. We don't want the drug to stuck in the stratum corneum or in the skin. We want the drug to go through the skin in a controlled manner and to reach steady state or pseudo steady state concentration in the plasma. That's what we want to achieve. The question is whether the model can answer this kind of questions and predict what will happen in those special populations. So what was the beginning? We had to understand the metabolism. What is really going on with selegiline, which appears in the body. So it's metabolized by, unfortunately, plethora of enzymes. So we had to really precisely define these metabolism processes, triggered by very different Cyps, as you can see here. And then these two chemically distant or different primary metabolites form a secondary metabolite, amphetamine, which we're interested in as well. So what were the steps of this exercise? The first one was the model building, the PPK model building, the basic model. So something what will allow us to understand whether the model works properly or not. Then model optimization, verification, and then utilization to try to answer the question which we asked. Can we predict a special population pharmacokinetics? Special populations I have in mind, adolescent population, 12 to 17 years old, as opposite to the adults, which were used for the model verification. Renal insufficiency and liver insufficiency defined by Child-Pugh scale. We're interested in those severely deficient patients at the level of the B and C of the Child-Pugh classification. So the first step, as I said, was the verification. So we developed the model and we ran the simulation and we compared the simulation versus the clinically observed data. So I'm just presenting 4 out of about 12 or 13 studies which we used at this stage. 2 for adults, healthy volunteer adults, and healthy volunteers elderly populations. And those dots are observed data, either average only, if that was the only data which were available, or together with the variability. And this gray area and the line here is an average prediction plus variability on top of this average prediction. So after running several studies, after running several simulations, we considered that the model is acceptably good. We can take it farther and we can utilize it. The verification was done not only based on the visual check. It's nice to have a look at the graph and to see whether it works or not, but obviously it has to be based on more quantitative metrics, if you like. So in this case, we compared the quality of the prediction for 2 parameters, PK parameters, Cmax and AUC, for all 4 moieties, chemical moieties, which we're interested in. And the acceptance criteria was 2-fold prediction. In most cases, the average value fit between 0.5 and 1.5 as for the fold of the difference between the observed data and simulated data. And then we took this model and we utilized this model in practice. So here what you can see, it's a very busy slide. It's not that important at the moment. Multiple clinical trials for special populations and the comparison versus observed data as for the PK parameters, Cmax and AUC. So as you can see, the prediction is acceptably good. So the conclusion was the model can be used for potentially dosing optimization, if there is a need for that, for special populations like liver insufficiency or renal insufficiency patients. And here is the beauty of PPK. That's what I love. Apart from being very practical, you can go back and by analyzing certain parameters, you can understand what influences the fate of your drug. So in this case, we analyzed multiple parameters and we asked the system the question, which of the parameter, the physiological parameter, really influences the pharmacokinetics of these drugs? Is it the amount of the enzymes? It is activity of the enzymes? Is it the age, binding, amount of proteins in the plasma, and so far and so on? You are able to do that with that, and that's the beauty. It's not that obvious based on the clinical trial. You've got limited amount of data which you can live with. Here, it's pretty unlimited. You can take whatever you want and you can use that. Very final slide, I promise. I'm finishing. Regardless of what I said and what I showed, it is all about mathematics. there is a mathematical equation behind this, nicely, hopefully looking graph. We obviously need experimental data, and the combination of these two gives us the possibility to understand the problem and to tackle the problem. PPK is there to help you, but obviously we have to collaborate. Experimentalists, clinicians, chemistry specialists, modelers, we have to collaborate. And the final message and sentence from my side is the scream for the collaboration. Let's do collaborate. That's the only way to push the science forward. Thank you so much. Thank you.
Thank you very much, Professor Polak. Unfortunately, the time is passing so quickly and we are quite late, but there is certainly time for at least one question. Yes, Professor Breimer.
Professor Polak, very impressive what you can achieve with this type of approach, and I appreciate, let me say, your emphasis on mathematical modeling and the enormous amount of data that you need to get reliable results. The only question I have is, how critical is actually the site of applications or the type of skin your ex vivo model for the ultimate result, for your predictability?
It is very important. Is that? Briefly speaking, or to make the long story very short, it is important. So depending on the place of application or area of application, you can have really different pharmacokinetics of the drugs. Yes. That's the one thing. In the model which I presented, we've got 9 different application areas: the head, trunk, arm, forearm, and so on, going down. And we run multiple studies comparing the, say, pharmacokinetics of the drugs, depending on where the drug is applied. There is a difference because the skin is different in different places of the body. So yes, it is important.
And can you thereby also suggest what the optimal site of application would be for a certain dosage form?
Well, if I had a model, good quality model for the drug of interest, I would say yes, it is possible. And that's what we are working on at the moment, actually, because sometimes some drugs are applied on the abdomen, and depending where specifically it is, the pharmacokinetics is different. So that's possible once you have a good quality model.
Sure.
Yeah.
Thank you very much for this fascinating news from this area of science. I'm sorry that we are half an hour late, forgive us, but I believe that science is passion, and passion also requires time. So we will continue, and we have still 2 short talks which are pre-recorded. And first one, the title is Personalized Medicine and Application of 3D Printing, FDM Technology in Pharmacy, and Marta Kozakiewicz, Anna Junak, Adrianna Złocińska, Bożena Karolewicz, Karol Nartowski. These are the authors, and the team comes from the Department of Drug Forms Technology, Faculty of Pharmacy in Wrocław, and also Laboratory of Elemental Analysis of Structural Research, also in Wrocław. So we'll continue.
Good morning, my name is Marta Kozakiewicz-Letała, and the topic of my presentation today is personalized medicine and application of 3D printing FDM technology in pharmacy. In a few initial slides, I will try to introduce to you the concept of 3D printing fused deposition method and the hot melt extrusion process required for it. Then I will present you the results of my study and the process of preparing tablets using the 3D printing method. The additive manufacturing process consists of 4 main steps, which are concept, hot melt extrusion process, computer-aided design project, and 3D printing process. And we can compare this schematic layered pyramid of additive manufacturing process to a layer printlet, where the first layer is the most important and determines the quality of a whole print. Here the same, the first layer or the base of the pyramid, which is the concept, decides on the success of the whole printing process. 3D printing technology is one of the rapid prototyping technique that uses the three-dimensional computer-aided design data to fabricate a scale model of a physical object. In this work, we use the fused deposition modeling method. This method uses the thermoplastic materials called filaments, which are heated in the nozzle of the printer and printed to the object layer by layer. Filaments are fed into the nozzle through the gear rolls at the top of the printer. But firstly, before the printing process, we need a design prepared in a 3D graphic design program of our tablets. Then our project is converted into the printer language called G-code, which contains all the parameters set by us, such as temperature, infill, and of course the printer movements. So what are the challenges for 3D printing of pharmaceuticals? The biggest challenge for wide application of 3D printing technologies in personalized medicine is lack of ready-to-use materials with pharmaceutical quality, which could easily be used in this process. Also, we have to mention the problem of miscibility of the drug, the polymer, and the plasticizer in the formulations that we are preparing. In this work, we designed the drug polymer plasticizer blends, which after processing using a hot melt extrusion process formed flexible filaments ready to use in the FDM-based 3D printing method. And to fabricate non-conventional filaments with incorporated drugs, we use a hot melt extrusion process. This is a continuous process using a single or a twin screw extruders like that one presented in the picture. The process is occurring under controlled conditions of temperature and the pressure, leading to obtain a homogeneous material. The process is carried out in the temperature range 50 to 60 degrees higher than the glass transition temperature or melting temperature of used materials. Plasticizers can be added to the extruder in two ways: directly to the physical mixture or pumping through the pump if the plasticizer is liquid. And why we should add the plasticizer? They are the key substance responsible for reducing the glass transition temperature of our material, the hardness of the material, and improving the flexibility of the filament, allowing it to be used in the 3D printing process. So during the process, we use the crystalline materials, which have a long range order and are characterized by the presence of sharp peaks visible in the powder diffractogram pattern, but also we use amorphous materials such as polymers, which can be defined by a short range molecular order and are characterized by a board halo. As you can see in the diagram, during the extrusion process, the crystalline drug and amorphous polymer are formed an amorphous solid dispersion, which they are mixed together at the molecular level. So we started our work with choosing a carrier for our API, and we chose the polyvinyl alcohol PATEC MXP with the glass transition temperature at 66.1 degrees. This PVA is dedicated to the fabrication by hot melt extrusion and amorphous solid dispersion because it exhibits high hydrogen bonding potential due to the numerous hydroxyl groups, which provide the ability to mix with API at the molecular level and stabilize API in the polymer matrix in amorphous form. For our plasticizer, we chose sorbitol. Due to good miscibility, we've chosen polymer. And for our model drug, we chose hydrochloric acid with a melting temperature at 268.6 degrees. So our concept was to combine substances with very different melting points and glass temperature transition, which needed to be processed by extrusion and then the 3D printing process under the same temperature conditions that make it possible to combine together but without thermal degradation, and that became a challenge for us. In order to evaluate the mechanical properties of prepared blends, we perform preformulation studies. We prepared blends with sorbitol and PVA at concentrations from 10 to 50%, and then melted them on a glass plate, mimicking the extrusion conditions. Powder X-ray diffraction analysis showed that the all prepared formulations were in amorphous state, as we can see by the presence of a bored halo on the diffraction pattern. We discarded the two blends with the highest plasticizer content that produced two viscous formulations. From the three placebo formulations, 10%, 20%, and 30%, we were able to form filaments, but only with 20% and 30% content of sorbitol. Filament was flexible enough, which allows us for the trouble-free printing of placebo tablets. Predicted in preformulation studies, tablets and filaments resemble a semi-crystalline nature of the obtained materials due to the presence of a bored halo in the diffraction pattern. Filament was planned with a diameter 1.75 millimeters to match with our 3D printer. And firstly, we prepared the filament only with hydrochloric acid and formulation of PVA with 20% sorbitol content, but the obtained filaments were brittle and breakable, which made them unsuitable for 3D printing. And the lack of the plasticity of the materials may have been due to the presence of hydrochloric acid in the crystalline form in the obtained filaments, which we can see on the diffraction pattern, the three sharp peaks. The melting point of hydrochloric acid is around 268.6 degrees, which is significantly higher than the extrusion process, 170 degrees, and may not have been sufficient for hydrochloric acid to completely melt and to transform to an amorphous state, causing filament brittleness. So the concept of combining two substances with a very different melting points, extruded at too low temperature for one of them, made it impossible to produce a sufficiently flexible filament. So in order to preserve the selected model drug, we had to decide how to modify our concept and to plasticize the obtained filament and also to adapt the API temperature to the processing condition of both hot melt extrusion and the 3D printing process. It was necessary to lower the melting point of hydrochloric acid. So to this end, an hydrochloric acid nicotinamide co-crystal with a melting point of 173.3 degrees was obtained, and this temperature was perfectly suited to an extrusion processing with a temperature of 170 degrees. So we linked high melting drug, hydrochloric acid, with a low melting conformer, nicotinamide, producing a crystal with desired melting temperature. This enabled us to fabricate flexible filaments and then print a series of hydrochlorothiazide co-crystal tablets. And we can see on the X-ray powder diffraction only broad halo in tablets and filaments without sharp peaks like before. So we can assume that the temperature of the process was enough to hydrochlorothiazide crystal to melt. For our dissolution studies, we used two methods. The first one was intrinsic dissolution, which is defined as the rate of dissolution of a pure substance under constant temperature, pH, and a surface area condition. We compare how the release of pure hydrochlorothiazide and the hydrochlorothiazide co-crystal that we made, which changed, and whether it could cause a modification of release. We can observe that the release profiles are not significantly different. However, we can notice a faster release of a hydrochlorothiazide from the co-crystal. And for printed tablets with co-crystal, we use the paddle dissolution as a characteristic dissolution method for tablets. And we can see on the diagram more than 50% of the drug dissolved in first 45 minutes, and after 90 minutes, more than 80% of the dose was released. Also, we can observe that 50% of the hydrochlorothiazide in interesting dissolution was released within 240 minutes. So the improved release in tablets may be due to the amorphous form of hydrochlorothiazide and the presence of the PDA. What is the conclusion? We could fabricate a non-conventional filament with incorporated API ready to use in FDM-based 3D printing methods, and through the modification of physicochemical properties of the API by fabrication and co-crystal enables us for successful processing of high melting point APIs. I would like to acknowledge Anna Junak for invaluable contribution in this work, Adrianna Złocińska for support in HPLC analysis, and Professor Karol Nartowski and Professor Bożena Karolewicz for supervising and guidance through the whole project. Also, I would like to thank Ministry of Science and Higher Education for financial support. Thank you very much for listening.
Thank you very much. Congratulations on your results, and I hope that Marta is among us online. So we'll have probably a question. Yes, there is a question in the audience. And I will take this opportunity to ask also those who are online, especially for tomorrow's session, just to write the questions or even to ask online.
Thank you very much for this very nice and interesting presentation. I'm just wondering that the approach to form a co-crystal is very nice, but I think it's probably easier to modify the extrusion process to increase the specific mechanical energy by increasing the temperature, changing the screw configuration. So did you try those approaches before you went this or added this additional layer of complexity to your work? Which I do really like, but I'm just curious whether it's not easier to modify the process.
Thank you for the question. Of course, it will be easier, but the temperature of melting point of the hydrochlorothiazide was around 260 degrees, and the maximum temperature of the 3D printing process is around 140 degrees. So it wasn't even possible to produce the filament with that high melting temperature. So we had to reduce it around 100 degrees. So producing eco-crystal was the only opportunity to use this substance in this process.
That is partly true. So I think it would be possible to increase the energy input during extrusion by modifying the screw configuration and the process settings to dissolve it even with this original high melting point. But this is maybe a different approach. It was very, very interesting, your results. Thank you very much.
Thank you.
There is another question. Yes.
Was the formulations that were prepared with, I mean, especially with the different quantity of plasticizer, assessed also for stability? I mean, the recrystallization potential in a long-term storage, because it's a known effect that the more plasticizers applied, where there is the more risk of the potential recrystallization of the amorphous drug over the long-term storage, like a couple of months. So was the stability tested for these models?
It's a very interesting question, but the printed tablets are really new, so we weren't able to check the co-crystal stabilization in our tablets. So it's for the further evaluation.
Okay, and is there any, well, also comparison between the co-crystals and the pure crystalline form of this API in terms of recrystallization potential? At least, well, the data that one or the other is more prone to recrystallization, or not really?
No, we didn't find it.
Okay, thank you.
Thank you very much, Marta Kozakiewicz. And we will go to the last our short talk. This will be Alice Biasin. The group will be also presented here at the audience by Beatrice Perusitti, because this is a collaboration between University of Trieste, University of Pavia, University of Ljubljana, and BIC in Trieste. Lipophilic ginger extract in controlled release tablets, pharmacokinetic and immunomodulatory effects. So please, we are looking for a lecture, yes.
Good afternoon, everybody. First of all, thanks to the organizers of this symposium for this opportunity. It's the first time for me that I speak in a congress. And today I would like to talk to you about a lipophilic ginger extract in controlled release tablet and its pharmacokinetics and immunomodulatory effects. Everyone knows about the ginger, an herbaceous plant with rhizome. This rhizome contains an oil resin in which we can find the gingerols and the shogals, the main bioactive constituents of ginger. In this figure, there are the molecular structures of the 6 gingerol and 6 shogal, the main representative molecules of these two classes of compounds. It's quite difficult to study the pharmacokinetic aspects of ginger compounds, because in literature there are conflicting data. The bioactive constituents of ginger are rapidly absorbed, but also extensively metabolized in humans. The liver and the intestinal microbiota take part in the metabolism of these compounds. The half-lives of ginger compounds and their metabolites in plasma are quite short. Besides, in many studies, it was not possible to detect 6 gingerol. His detection depends on the dosage and on the type of the ginger extract. To bypass these pharmacokinetic problems, In our pilot study, we consider an innovative lipophilic ginger extract obtained with a supercritical food extraction. We consider a lipophilic extract because of the lipophilic nature of the bioactive ginger compounds. In particular, we consider a ginger extract titrated at 17% in 6 gingerol and 5% in 6 sugar. 100 milligrams of this extract was formulated as fast-release soft gel capsules and also as slow-release tablets. We want to compare these two different formulations to evaluate if the slow-release tablet could bring some advantages in front of the particular pharmacokinetics of ginger compounds. For the pharmacokinetic study, a soft gel capsule was given orally to 8 healthy volunteers, and after 1 month, a slow-release tablet was given orally to the same 8 healthy volunteers, and blood samples were collected at different time points. Regarding the slow-release tablet composition, in this slide are reported the excipients. The slow-release system consists in a hypromellose-based erodible matrix, and the tablets were obtained with a direct compression method. To study the slow-release system, we have taken an in vitro dissolution test. In this slide, we have the in vitro dissolution profile of the slow-release tablets, in which we have the percentage of 6 gingerol and 6 sugar released by the slow-release tablets versus time. As we can see, the 80% of the bioactive compounds were released within 12 hours. This profile indicates a constant rate of release, and this profile is conventional for a slow-release formulation with an erodible polymeric matrix. As such, the dissolution test is a first confirmation that the tablets are able to control the release of the phytocomplex. After the in vitro dissolution test, we have verified the in vivo pharmacokinetic performance of the two formulations, the soft gel capsules and the slow-release tablets, both containing 100 mg of the extract corresponding to 70 mg of 6 gingerol and 5 mg of 6 sugar. In this slide, we have the plasma concentration profile for the soft gel capsule assumption that shows that 6 sugar is absorbed rapidly and detected in plasma after 35 minutes, in agreement with the lipophilicity of the molecule. The 6 sugar glucuronide is detected in plasma before 6 sugar, indicating that the conversion to glucuronide is very rapid and that 6 sugar glucuronide is pre-systemically formed. Furthermore, the plasma concentration profile for the soft gel capsules displays 2 peaks for both 6 gingerol, 6 sugar, and 6 sugar glucuronide. So, it can be hypothesized that the lipophilic extract of ginger undergoes to enter hepatic circulation. Then we have considered the pharmacokinetic performance for the slow-release tablets. In this slide, we have the plasma concentration profile for the modified release tablets that exhibits a first enterohepatic metabolism for 6 ginger, for 6 sugar, and then a constant blood concentration of 6 sugar from 4 to 12 hours after the oral assumption. This plateau is possibly due to the control-release matrix system, which modulates the release of the ginger extract and its active ingredients. Interesting, for both the oral formulations, 6 gingerol and the glucuronide conjugate were not detected in any plasma samples, probably because of the conversion between 6 gingerol and 6 sugar in the gastrointestinal fluids, and probably because of the pre-systemic metabolism. In another study, other researchers of my group have noticed that combining ginger extract with another vegetal extract containing surfactants, it was possible to detect 6 gingerol in plasma samples, probably because of the formation of micelles containing 6 gingerol. Now we can consider the pharmacokinetic parameters. Here are reported the pharmacokinetic parameters calculated with a non-compartmental approach. To better evaluate this data, I'll show you the corresponding graphics. There are statistically significant differences between these parameters, such as for maximum time and area under the curve. According to this data, it can be speculated that the presence in the systemic circulation of 6-sugar and its conjugate metabolite coming from slow-release tablets is longer lasting than those coming from the fast-release capsules. So it is possible to confirm the validity of the control-release matrix system. Another purpose of this study was to evaluate the immunomodulatory effects of the ginger extract, formulated in slow-release tablets. In this author, I reported many pharmacological properties for ginger and sugars, such as anti-inflammatory and immunomodulatory actions. These properties seem to be a sum of different mechanisms of action. In vitro studies suggest that gingers and sugars can inhibit prostaglandins, leukotriene synthesis, and reduce nitric oxide and pro-inflammatory cytokines production. There are some evidence that probably these anti-inflammatory actions are due to the downregulation of gene expression, such as regulated by the transcription factor NFKB. In this study, the evaluation of the immunomodulatory effects of ginger extract was made by gene expression profiling. A single slow-release tablet was administered to 8 healthy volunteers. Blood samples were collected at baseline, time 0, and after 24 hours from the administration, time 24. The total RNA was extracted from peripheral blood mononuclear cells in order to conduct transcriptional analysis, such as NGS sequencing and subsequent gene set enrichment analysis, and real-time quantitative PCR. The gene set enrichment analysis allowed to establish which kind of pathways are differently transcribed in our cell samples with respect to the control. In our study, the controls were the time 0 samples, that are the samples collected before the oral assumption of the extract. Analyzing RNA sequencing data with the gene set enrichment analysis, we have found that some inflammatory pathways and some immune system pathways stand out as statistically significant for each subject. These 8 subjects are the 8 healthy volunteers. In other words, some inflammatory genes and some immune system genes were differently transcribed after the ginger assumption in each subject. We have also made another gene set enrichment analysis, where the gene sets considered were identified in a previous study conducted on the transcriptional regulation after the systemic administration of hydrocortisone in 2 different doses, 50 or 250 milligrams. Interesting, for 4 subjects, these 4, we have found that there is a statistically significant correlation between the gene sets that are differently transcribed after systemic assumption of hydrocortisone and the gene sets that are differently transcribed after the oral assumption of ginger extract. So there is a correlation between the genes modulated from hydrocortisone and the genes modulated from gene. Based on the results of gene set enrichment analysis, we have also paid attention on the expression of single genes correlated with the inflammatory pathways. We have considered the expression both on the RNA sequencing data and also on the real-time quantitative PCR data. Indeed, real-time quantitative PCR data is a more quantitative technology. In this slide, we have the levels of expression from some gene. In the upper side, we have the box plot with the RNA sequencing data. And in the lower side, we have the box plot with the real-time quantitative PCR data for the same genes, such as interleukin-1 beta, interleukin-6, and so on. For each gene considered, we have the levels of expression at time 0, blue box plot, and at time 24, yellow box plot. That is the time after the ginger assumption. We have studied different genes, but I reported here for brevity only interleukin-1 beta, interleukin-6, histidine decarboxylase, and interleukin-10. We can see an agreement in the expression levels for these genes between RNA sequencing data and real-time quantitative PCR data. And And this is a confirmation for the validity of RNA sequencing data. We have noticed a downregulation for time 24 samples for all the pro-inflammatory genes, such as interleukin-1 beta, interleukin-6, and histidine carboxylase. Instead, for interleukin-10, an anti-inflammatory gene, we have an upregulation in time 24 samples. So, for the pro-inflammatory cytokines that we have considered, we have seen a statistically significant downregulation. So, in conclusion, the slow-release matrix system modulates in vitro and in vivo pharmacokinetics of the ginger phytocomplex components. Furthermore, the gene set enrichment analysis highlights how the lipophilic ginger extract can modulate the transcription of functional genes connected to the immune system and the inflammatory response. We have also seen that the ginger extract can globally decrease the expression of pro-inflammatory genes in peripheral blood mononuclear cells. We are also working for the comparison of the immunomodulation effects between the slow-release tablets and the softgel capsules. So this is all. I would like to thank Professor Luigi Macchioni and his group, my thesis supervisors, Professor Dario Vajnovic and Dr. Roberto Berardo, Professors Beatrice Perissutti and Luton Asa, and the Department of Chemical and Pharmaceutical Sciences of the University of Trieste for funding. And thank you all for your attention.
Thank you very much for these impressive also results. And maybe there are questions. Yes, Professor Breimer.
Very interesting talk. How stable, actually, how consistent is the composition of the ginger extract? So in other words, the extract, of course, may vary in composition, or can you actually make it comparable all the time?
Can you repeat? Sorry.
How consistent is the composition of the ginger extract? Because natural products usually vary in composition.
Did you standardize?
Yes.
Yes. Our lipophilic extract was titrated at 6 gingerol and 6 shogol, and also there are other components that are lipophilic components, and we have also little amounts of other ginger components bioactive that are such as paradols. They can form themselves in the stock of this extract.
Thank you, and nice to see you on the screen.
Because we are late, so we will close this session. Thank you very much, Elise.
Thank you.
Once again. And we are sorry for being late, and we invite everybody tomorrow 8:30. We'll meet for Friday sessions. Thank you very much. Goodbye.